Hi, folks. We're going to go ahead and get started. My name is Judy Morrow. Welcome to the Pharmacovigilance and Signal Detection course at ICPE All Access 2021. What you see on your screen today is our agenda for the morning. We're going to start out with a little bit of just housekeeping. Then Greg, who you see as one of your instructors on the screen, is going to be giving the first lecture on the introduction to pharmacovigilance. Then Nicholas is going to be giving the second lecture on statistical pattern discovery and pharmacovigilance and its pitfalls. We're going to go ahead and take a coffee or water break for 10 minutes. Everybody can get up and stretch. And then I'll be giving a talk on novel quantitative methods for identifying drug safety signals in electronic healthcare data. With that, we have a signal detection live exercise. I've put a link in the chat, which I'll repeat because I think people who join the chat after the link is posted may not have access to it. And then Monica will close us out with pharmacovigilance in practice, getting from signal to insight. I'm going to ask each of the speakers to do a brief introduction of themselves before they speak, so everybody knows sort of where they're coming from and the experience that they bring to the course today. We'll end with some Q&A, and with a small crowd today, we'll likely be able to have everybody unmute and be able to ask questions at will. But if throughout the course you have questions, we can get those put in the question and answer box, the chat box. And so here's just an information about the materials today. So the course directors, what I mean by that is the ICPE directors themselves were kind enough to create a Google Drive link for people who have access to Google Drive, and you can get the materials that way. I've put the link again in the chat. So during our talk today, there's going to be a chance for sort of a live demo where you can follow along and do a signal detection exercise at home. But to do that, you need some materials. So one is a computer program that you can download onto your computer. It's available for PC, Mac, and Linux operating systems. And then there's a couple of handouts to go with it. These two files are available there. Just to note that even though the ending looks funny, it's a text file. So if your computer is mad about you trying to put it on your machine, just know that it's a text file. And then additionally, there is a PDF of all of the course materials for today that's available at this site. So just some brief requests. Please limit your questions during presentations to clarifications, and please go ahead and type those in the chat box. We will try to make sure that we stop and get clarifications as we go, but we hope to save the larger commentary and discussion for the question and answer session at the end. Just to note that all opinions expressed in these lectures are those of the presenter and do not necessarily represent the views of their parent organizations. We all come from different parent organizations, and I think that's true of all of us. And then finally, a note that terminology in this space is not uniform across international jurisdictions, but we're trying hard to bridge those barriers and to strive to communicate the intent behind the terms we use, acknowledging that these concepts may be named differently in different settings. And we'll try to do our best to use all the terms that might be appropriate in any given situation. And so with that, I'm going to go ahead and stop sharing and turn it over to Greg for our first lecture.
Okay, thank you, Judy. So hopefully my slides are coming up. Can you see my slides?
We see them.
Great, that's a successful meeting already. So I am Greg Powell. I am a pharmacist by training. I have been at GSK now 21 years. The entire time has been in the safety group, and I've always have worn two hats. On one side, I've been a safety evaluator. I've done just about everything you can do in drug safety, from phase 1 studies, phase 2, phase 3, post-marketing registries. And then the second part of my job, since I've been at GSK, is really also focusing on advancing the science of pharmacovigilance, looking at new data sources, new techniques, really how we can do the best job we can with the data we have to protect our patients. As far as disclosures, the opinions expressed are mine. I am a full-time employee of GSK, and I will talk about some tools within my presentation that GSK helped co-develop. GSK received, and nor myself, received any financial benefits from these tools that are commercially available. I will primarily focus on general post-marketing signal management today. There's a lot more to pharmacovigilance than just post-marketing. I will briefly talk on non-post-marketing data sources, but the bulk of my presentation will be spent on post-marketing signal detection. And also, just to acknowledge, I am not an epidemiologist. So if I misuse any epi terms, please forgive me, because I have not taken the first class in epidemiology. Also, I will share along the way, I will share some personal experiences with the various data sources. These are unique to me. They may or may not be generalizable to other drugs at my company or even outside of GSK, but it's always nice to kind of see where rubber meets the road, and just some practical insights into how this actually is operationalized. As far as nomenclature, as Judy mentioned, we all have there's lots of different definitions for the same term. The bulk of what we use comes from the EMA as far as the definitions. Pharmacovigilance itself is the science and activities relating to the detection, assessment, understanding, and prevention of adverse events. A signal is essentially information that suggests a new or changed association between a drug and event that is judged to be a sufficient likelihood to justify verificatory actions. Talk today. And then really everything we do with a signal, from the time we detect it to the time we assess it, all that's wrapped in under one umbrella called signal management. So the purposes, for this lecture, signal detection is defined as the process of really looking for any drug event pairs that we want to investigate further. We use quantitative methods, and we also use qualitative methods. So anytime a quantitative threshold is crossed or a qualitative alert or qualitative criteria is met, we call that an alert. Once we've identified an alert, we will look into the drug event pair a little bit deeper to see if we can actually validate the drug condition pair and really assess if it does require additional work or not. Of the signals that are validated, then we progress to signal assessment. And this is really where rubber meets the road, in that we look at the totality of the data and really try to determine if there's a reasonable chance of a causal association or not. So with that, defining the terminologies that I will use through the presentation, an outline is I'm going to spend a little bit of time talking about pharmacovigilant workflows. But the bulk of my time by far will be spent really on the various data sources that we use in pharmacovigilance, because everything we do in pharmacovigilance and drug safety starts with data. Data is the very foundation that we build our safety activities upon. And then I will briefly introduce the concepts of signal detection, both qualitative and quantitative, but Judy and Nicholas will go into far more detail on those. Looking at everything, why is post-marketing safety surveillance so important? We do a lot of clinical development work. Many of our drugs have been in clinical trials for years. So why, is post-marketing safety surveillance so important? And the reality is, when a drug is launched, we have a really good idea of the efficacy. We have a really good idea of the vast majority of the safety piece, but we don't know everything as it relates to safety. So there is still some things to learn long after a drug has been launched. And there's been some real notable examples in the post-marketing setting where significant safety things have happened. So a couple of the examples I have here, includes sulfanilamide, where something is simply trying to go from an oral tablet and powder formulation to a liquid formulation. Unfortunately, they dissolved it in diethylene glycol, which is antifreeze, which resulted in over 100 deaths. Thalidomide, which a lot of people here probably have heard many times about, a drug that was originally marketed as morning sickness in pregnant women, that has been shown to resulted in somewhere between 10 and 20,000 birth defects. Other examples of notable safety signals identified in the post-marketing setting include diethylstilbestrol, which after 37 years on the market was shown to be associated with various types of cancer. Fen-phen was on the market for 24 years. It was a diet drug that wind up getting pulled off the market due to heart problems and pulmonary issues. Regulin, which was off the market after 3 years due to hepatotoxicity. And the one that probably most people are most familiar with is Vioxx, which was recalled after 5 years on the market. So at the end of the day, post-marketing safety surveillance is really important. If you look at the CDC, they estimate somewhere between or approximately 1.3 million ER visits in the US each year are attributed to adverse events, and somewhere around 350,000 hospitalizations. So there's a lot of people in the US, there's a lot of wonderful medications on the market. There are no medications that are perfect, meaning there's no adverse event profile. And these adverse events do lead to hospitalizations. They unfortunately do lead to death. So understanding how a drug and the safety profile, even post-marketing, is critically important to maximizing the benefit of the products. So what is pharmacovigilance? The goal, as I gave you the definition, but really the goal is to identify safety risks as early as possible in the product life cycle and characterize the risks so that we can identify appropriate risk mitigation activities. When it comes to risk in pharmacovigilance and drugs, part of the risks are diversifiable, part of the risks are not diversifiable. So we really want to be able to fine tune where we can to really maximize the efficacy, minimize the harm, and really try to offer as much value and as much benefit as we can to our patients. Traditional PV activities, if you had to group them into buckets, really they fall into 4 buckets. Signal detection, which I've mentioned in the definitions, which is hypothesis generating. Signal strengthening or validation, that's where we take the signal, look at, and really try to ensure it's something that warrants further investigation. A signal assessment or hypothesis testing is where we go in and confirm that either there is a reasonable causal association or not. And I think it's important here to note that signal assessment or hypothesis testing, sometimes this may be an epi study where there's a formal statistical test. Many times it's actually not, where it's just purely a qualitative clinical evaluation of the data, and we make a judgment call. And then the 4th bucket is around risk mitigation. How do we communicate risk, add it to the label, but also what additional activities that would we need, whether it be registries, limited distribution, things like that. And then everything we do in pharmacovigilance, it starts with the data, as I mentioned before, and I'll probably mention a couple of times. The data is the foundation of what we do. We apply various techniques and things that Judy and Nicholas and others will talk about, how we turn the data actually into information that we can use. Then, of course, once we have the information, then we turn that information into insights, which allows us to make decisions about the safety of our products. There's lots of sources of data that we use. As I mentioned a couple times now, data is the foundation of what we do. There is no perfect data source. All data sources have strengths, all data sources have limitations. And really the importance of a particular data source really depends on the drug event pair of interest. Depending on the attributes of the drug, the attributes of the event, that to some way helps us guide us into which data source may offer the most value for that. And in some cases, it may be more than one data source as well. So as far as getting into the data sources that we use in pharmacovigilance, I'll briefly touch on preclinical. And what preclinical data is, I've broken it into 2 primary buckets. This is what I call test tube data, where we have various assays that we can run in the lab to test the drug itself. For example, we can have a petri dish of liver cells and see what the drug might do in the liver. And then we also do in vivo testing or animal testing as well. The advantages of preclinical data is that this gives us a starting point to safety. Whenever we do the animal testing, and the in vitro testing, it really gives us an idea of what some of the risks may be, and it gives us a starting point to what we may see in humans. During this preclinical testing, we actually start developing a risk table where it may be theoretical risk based on some of the results, or identified risk based on the pharmacology of the drug. But it really gives us a starting point of what we will see before the drug ever goes into the first human. It also gives us some idea about dose relationship. Within the animal testing, and in vitro to some degree, there is a range of doses that are tested. And what we do is we see during the dose escalation to see at what level do our adverse events starting to occur. And we call that the no adverse event level. And that's really important for us. So for example, if the no adverse event level in an animal is 100 milligrams per kilogram, when we go into humans, if we start with a human dose of 10 milligrams per kilogram, that gives us a factor of 10 protection, if you will, meaning that the dose we're going to humans is 1/10 the dose that the minimum level required to see adverse events in animals. So it gives us, if you will, a degree of safety coverage. Ideally, the human dose would be far below the dose that we see adverse events in animals. But of course, that's highly dependent upon the drug itself. For an antihistamine, we want a huge amount of tox coverage, meaning a large difference between where we saw adverse events in animals and what we dosed with humans. But if this were a cancer treatment, we may be more tolerant to actually that being the same level. And also what's nice in animal models is that we do get a degree of reversibility as well. If we do see an adverse event, once the drug is stopped, does the adverse event go away? And that's really important because once we get into humans, if we start seeing something, knowing that if we stop the drug and the adverse events likely goes away is really, really important in protecting our patients and our subjects in our trials. But of course, preclinical data has lots of limitations, like all data sources. Preclinical studies are not large. There's a high degree of variability as far as positive predictive value between animal in vitro testing and humans. Humans aren't rats, humans aren't monkeys, humans aren't dogs. So although we may see it in a certain species, there's no guarantee that we may see that in a human, and we may see something in humans that we did not see in animals. And of course, no preclinical data is even remotely related to real-world context of how the drug's going to be used. Moving on to the next data source, clinical trials. Everybody here knows what a clinical trial is. I'm not going to go into a lot of details, but really this is probably the best data source that we have with respect to drug safety, because these are controlled environments. We have, we know exactly, well, not exactly, we know most of the variables going on with the patients. We have strict inclusion-exclusionary criteria. We have continual monitoring throughout trials. We're routinely engaged with the investigators. So if we see something, we can actually discuss with them if additional tests are needed, what treatments are needed. So really, clinical trials are the gold standard when it comes to all data sources, and especially within adverse events that are identified during clinical trials. But it's important to realize that, as I mentioned a few slides ago, there's still a lot of adverse events that we identify outside of clinical trials in the post-marketing setting. Key limitations to clinical trials data include generalizability to the real world. Relatively speaking, these are small sample sizes, and this is a really key point. A large clinical trial may have 2,000 or 3,000 subjects. So if you're worried about Stevens-Johnson syndrome, progressive multifocal leukoencephalopathy, or something that may be 1 in 10,000, 1 in 100,000, statistically speaking, you may not see any of those in the clinical trials, and those may not manifest into the real world use of the drug, where your sample sizes are well into the millions. And generally speaking, clinical trials, a long clinical trial may be 1 or 2 years of exposure, where in the real world, people will take some drugs for the rest of their lives. So long-term exposure is also a limitation of clinical trial data. So now moving on to post-marketing spontaneous data. And essentially, a spontaneous report is any unsolicited communication that describes one or more adverse drug reactions. This is the bread and butter of post-marketing safety surveillance. This is one of the worst data sources you can imagine. But in the post-marketing setting, it's also the best data source that we have currently, given the breadth and depth of its coverage. And also that spontaneous data, its primary use is to report adverse events, where some of the other data sources we use are more secondary in nature. So people love to talk about how bad the data is in spontaneous, but to date, other than regulatory communications, this is the largest source of post-marketing safety signals that we have in the industry. So in order to be considered a spontaneous report, there are 4 basic elements that are required. To be a spontaneous report, all we need is an identifiable patient, identifiable reporter, an identifiable drug, and an identifiable event using the broadest definition possible. So if I called a pharmaceutical company and said, hey, I have a patient taking atenolol and they had a rash. Those are the 4 elements because I'm a reporter. They don't even need to know my name. I said I have a patient, which is identifiable, having a drug, an event. And a report like that, and you'd be surprised how many reports contain that little information, then we're required to review and to assess causality without any additional information whatsoever. So the strengths of spontaneous data include that this is coming from the real world. There's lots of bias, there's underreporting, lots of limitations on the next slide, but it's still coming, it's more representative of real world use than clinical trial data. You get much larger exposures than we do with clinical trials. A typical pharmaceutical company can receive hundreds of thousands of spontaneous reports a year. And because of the numbers, you can actually start seeing some of those more rare adverse events, things that, PML, SJS, things that really keep us awake at night, but are so rare that we may not see in clinical trials. As far as limitations, the first thing is there's no unknown numerator. There's several studies out there that suggest that greater than 95% of all adverse events never get reported. And those that do there's certainly a fair amount of channeling bias. It may be being reported because of a litigation issue. It may be reported because a famous person on TV mentioned they had this event. But nevertheless, most adverse events never get reported. So we really don't have a numerator. And we also don't really have an exact denominator. We can look at sales data and approximate how many people have been exposed, but it is a best guess when it comes to the denominator as well. The lack of quality, we actually did it recently. We did a project and we evaluated a sample of spontaneous adverse events. And with the initial report of the 26 data elements that could be considered important for assessing causality, the initial reports contained 13 of the 26 data elements. On most of our adverse events, if where possible, we do try to get follow-up information to add to the quality. But what we found was that only 13% of all adverse event reports will even receive follow-up information. And on average, the number of buckets of valuable information go from 13 to 15 out of the 26. So even with throwing all that together, we don't get a lot of fantastic quality within spontaneous. It's highly variable. Interesting enough, sometimes you'll find certain disease states the reports are better than others. With some of our HIV products, some of the best spontaneous data I've ever seen, people who are more engaged in their care and report adverse events tend to give better data when it comes to reporting an adverse event. GSK did co-develop one of the leading tools to evaluate quantitatively spontaneous data. And what we found was first of all, I think just to realize the magnitude, GSK has about 800 or so drugs. There are roughly 20,000 or so different MedDRA-PTs within the database. So on average, there's about 18 million possible drug event pairs that we have to look at. So there's a massive amount of data coming in, massive amounts of things that we have to weed through. You do see a lot of drug event pairs related to the indication. Some of the highest signal scores you see will be related to indication. You see some non-specific drug event pairs. So really at the end of the day there's a lot of data coming into this. The data, spontaneous data, although I wish it were a lot better, quality is still the gold standard in post-marketing safety surveillance. And in my opinion, likely will continue to be for a long time. The second data source that we use in post-marketing surveillance is the published literature. We routinely review abstracts from all journals, meeting abstracts, anything that's published. And really we review the literature based on from two perspectives. One, we identify articles that contain the 4 data elements that meet a spontaneous adverse event definition. Therefore, we would enter it into our database, we would send it to the regulatory agencies. But we also review every literature article within the context of what impact does this have on the drug, on the benefit risk profile of the drug. We may see a rash reported with Augmentin, and we have to report that, but rash is well characterized as Augmentin. So although it's an adverse event, it really doesn't change the benefit risk profile because it's a well-known and well-characterized risk already. Things in the published literature as far as strengths, certainly peer reviewed. Generally speaking, you have very high quality data in the published literature. And generally, the authors do a really good job of contextualizing, trying to assess other variables, other potential causes, comorbidities, concomitant medications, really good information. But one of the significant limitations of the literature is by the time a physician sees 3 or 4 interesting cases, they document it, they write an article to the New England Journal or somewhere like that. Many times that can take years. And if you go back and think about what one of the goals of pharmacovigilance is, we want to identify drug condition pairs as early as possible in the product life cycle. So although the published literature is fantastic data, many times there's years between somebody experiencing an adverse event and by the time it comes out in print. As far as having reviewed many thousands of these myself, what you find is most of the articles fall into 1 or 2 buckets. Either one, very anecdotal experience describing 1 or 2 cases of something interesting, an interesting event, or the vast majority is actually, we see a lot of preclinical testing where somebody does a rat model, even with older drugs. One of the drugs I used to be responsible for is salbutamol or albuterol. And even to this day, that drug was approved in 1968. You still see a fairly large number of people using it in various animal models and publishing the results as it relates to a wide range of topics. And then going back to what I mentioned about preclinical, the correlation between an animal model and a human model is highly variable, and many times it's not as predictive as we would like. So moving on to, I'm sure everyone's favorite data source, observational data. When I say observational, I refer to medical records, entrance claims databases. And again, you guys could teach me about this stuff. From my use of observational data, these are fantastic and I would say underutilized even to this day. These are very large databases. Some of the claims databases I've worked with are well over 100 million patients in nature. The longitudinal nature is fantastic. We get to see what happened before the event, during the event, and potentially after the event for a subset of the patients. That longitudinal nature, even just having a 2 to 3 year snapshot is fantastic because so much about drug safety is about context. You need to contextualize the drug itself, you need to contextualize the event, and then contextualize the patient who may be receiving the drug. And it's really the intersection of those 3 is how you really go about assessing a potential causal association. As far as limitations, you guys could teach me probably hours on the limitations of the data, so I'm not going to go into the limitations of observational data just because that's not my area of expertise and I don't want to say something really silly. As far as real world use, we did actually a number of years ago develop a tool that allowed us to do signal detection and signal detection and observational data, and also had rapid query capabilities. And from our real world experience, what we found was that by far, at least the way we utilized it, the most valuable feature was the rapid query capabilities. If I'm doing a safety assessment and I'm evaluating hepatotoxicity, it's fantastic. And within a matter of minutes or hours, very rapidly, you can actually look at, okay, what % of people who take this drug may have a history of hepatitis? What % of patients who take this drug may have a history of taking statins or something that may be another hepatotoxic drug? The rapid query capabilities are fantastic because what it allows us to do, those of us with clinical backgrounds, we've seen patients, we have a lot of intuition what may be going on in the medical context, but this allows using empiric evidence to really quantify exactly what's going on there. So the rapid query capabilities are fantastic. When it comes to use for general signal detection, we had kind of a mixed bag with ours. Part of it was the challenges of not all, as I mentioned before, we have over 20,000 measured terms. Well, not all of those will have a corresponding ICD-10 code. And I'm not going into too much detail here because I believe Judy will touch on this a little bit more. So our experience was, rapid query capability is fantastic. And there's absolutely value, I think, in the signal detection side. It's just really there's still some maturation in that process to better understand how it fits in. As far as emerging data sources, the one that's been published on quite a bit here recently is social media. And when I say social media, essentially people always throw social media all into the same bucket. In reality, not all social media is the same. Somebody who posts a little snippet on Twitter and somebody who gives you their life story on Facebook, those are both social media, but the value of the data is very different. We've done a lot of original initial research just trying to assess where social media can fit into our pharmacovigilance toolkit. And what we found was, the good news about social media is there's a large amount of data. In 1 year, there are more adverse events discussed in social media than in the entire FDA's database. Huge amount of information. People are also willing to discuss topics that they wouldn't normally discuss with a pharmaceutical company, a regulator, or their physician, and specifically around drug abuse and misuse. You do get geographic diversity within social media. Just about everybody on the planet, I think, now has a Facebook or Twitter account. So you get some geographic diversity. And one thing here that we borrowed learning from observational data, one of the things we found to unlocking the value of social media is actually looking at longitudinal posting records. So rather than just look at a single post, because for a chronic disease, it's rare you're going to get all the information in one post. But if you link posts together, and we de-identify these before we get it, so link the post together, de-identify it. When you get that disease journey or the journey with using a drug, you get a much more complete picture of what's going on, a higher likelihood of getting con meds, comorbidities, things like that. As great as that sounds with social media, I would say the weaknesses are equally as daunting as well. First, on social media, the validity of the data. People lie, people exaggerate, machines can post social media posts. I mean, it's the wild, wild west of data sources. It's probably, as far as the quality of the data, it's probably worse, generally speaking, than spontaneous data, because you get everything. You do get a lot of duplications of posts across social media channels. The average person, I think, has somewhere in the neighborhood of 6 or 7 different social media accounts. So if they talk about, their experience on YouTube, they may also post on Facebook. They may also post it on Twitter. You do get limited medical context. Unhappy. There have been some studies showing that unhappy people post more frequently than happy people. And also what is posted online is there's some interesting work that's been done where if a celebrity would come out and say they had a drug and had an adverse event, it's amazing how that peak goes, you see that carried over into social media, or a legal advertisement or advertisement about, did you take a drug, were you harmed? You see those will actually dramatically stimulate discussions within social media themselves. So social media itself is we're still not quite sure what to do with it. There was as far as real world examples, I was part of the IMI Web Radar project where we evaluated the value of social media. And what we came up with is there's so much noise in social media. And going back to the size of the social media, although social media is probably the largest repository of adverse event discussions, the number one most frequently discussed adverse event is my flu shot hurts when I get the shot. The second most common one we looked at it was Benadryl or diphenhydramine makes me sleepy. So although there's a large quantity, the vast majority of the quantity is about, say, articulating adverse events that are well known, well established with the drugs. It's still trying to decide the value of using social media as to identify signals or emerging safety issues that maybe aren't well known, well characterized. So in our Web Radar paper, basically what we summarized was that social media is still too early to be a mainstream tool for post-marketing safety surveillance. There are some niche areas, however, where it offers a lot of promise. We published a paper and another company's published a paper about when it comes to abuse and misuse, I would argue that probably social media is the best data source. But outside of that one niche area, we still have a lot to learn to, where does this fit? It's such a large data source, we can't ignore it. But where does it fit into, the different tools we have, and how can we use it to maximize its value and minimize the amount of noise that it causes in the pharmacovigilance process? Another emerging data source is crowdsourcing. Those of you who are not familiar with crowdsourcing, essentially crowdsourcing is Uber for intellectual thought. We've done some work in this area, and there's some general crowds such as Amazon Turk, where you can go in and directly interact with people, ask them about adverse events specifically, Or you can go to a physician crowdsourcing network, one called Sermo, where you can go in and actually ask physicians directly about their experiences with the drug, adverse events. The advantages of crowdsourcing over something like social media is there is an interactive component. Unlike social media, where it's primarily social listening, whatever's written, we just take at face value. Here, we have the ability to direction the discussions. We can ask for follow-up information. We have the ability to really just get better data than we would from social media. But of course, there's still some limitations in this very early days right now with respect to social media, as far as the data validity. How do we know people are telling us the truth when we crowdsource issue topics? And also, people must have internet access. What we've used crowdsourcing for right now is we use it to develop training data sets for some of our machine learning and artificial intelligence work we're doing. It actually works really well. If you have a large data set and you want to establish the known truths, true positives, this is fantastic. We get very large data sets created very quickly to help feed into the algorithms. But we also did this to assess, and this manuscript should be coming out soon, perceptions of a pregnancy registry we have. What are barriers to the pregnancy registry? Why are you using it? Why you're not using it? So this is still even earlier days than or earlier days in social media data, but it's still something on the horizon that may be an appropriate tool in a pharmacovigilance toolkit. Getting even further out from mainstream PV is the genetics. Certainly a lot of information about genetics coming out, and we're still in the early days of trying to figure out how that can fit into drug safety. The nice thing about genetics, we do find a genetic basis for an adverse event, then that's a very strong correlation because it can be predictable, and it gives us more of a chance to also maybe, in a risk management perspective, manage that risk, whether it be, do some genetic testing and to help, and if they're predisposed to an adverse event, maybe using another treatment, or at least do some more intensive monitoring. But the challenge is there's so much information about the DNA now. There's so many adverse events. There's potentially, I'll talk about 18 million drug event pairs and spontaneous data. You could even multiply that by a very large number here. There's just a huge amount of data and potentially a huge amount of noise in this area. But nevertheless, there's a few different examples where genetics can be used to predict adverse events. There's one around, there's a fair amount of genetic variability in the liver and how the liver metabolizes drugs. So there's a number of articles about how that can affect the pharmacokinetic profile of a drug. There's a genetic link to warfarin variability. Most warfarin patients fall into those that are very stable, or those that I used to call brittle, meaning their INR values are all over the place. They've identified a genetic link to that, and you can actually test to see if a person's maybe more stable on warfarin, or if they have higher variability, then maybe that patient that you would want to use maybe one of the antithrombin 3 inhibitors on. And then there's using the GSK very early on, there's some experience with genetics and abacavir hypersensitivity as well. So still trying to figure out what to do with genetics. And then finally, the most current thing, and in some ways the coolest thing, but also the thing that scares me quite a bit, is the Internet of Things. Everybody has a cell phone, everybody has internet access. Your watch talks to your television, which talks to your refrigerator. Everything's eventually going to be connected. How can we leverage that for drug safety? The advantages of something like this is you get fantastic geographic coverage. Everything's connected to everything. You do have the ability for some of these to interact with the individual, but the limitations of the Internet of Things, how do we use this? There's a lack of standards across the different connected devices. There's security, privacy concerns. There's data storage, data retrieval. As you can imagine, even more so than DNA, there's almost an infinite amount of data points that can be generated through electronics. How do we go through an infinite number of data to really distill it down to those medical insights that we need to protect our patients? There are some examples. We did a collaboration with Propeller Health, and Propeller Health has done some work around use of geotagging use of albuterol. So you can look for geographic patterns of where people are using their emergency or their rescue inhaler to see, is there a certain park or is there a certain place that exacerbates asthma more than others? So there's still a lot to do. I think there are some Internet of Things devices out there, but I think, this area is about to explode over the next 5 or 10 years. So in summary, there's lots of data sources that have lots of uses. Generally speaking, for signal detection, spontaneous and literature reports are our two primary sources in my experience. When it comes to signal strengthening and signal assessments, that's usually where we dive into the observational data, really leveraging the value that observational data offers. With social media and the other data sources, there's still really to be determined how it fits into our pharmacovigilance toolkit. Social media, certainly abuse and misuse is one area, but is there something beyond that? We're not sure yet. And I think it's important to acknowledge within signal assessment, really in signal assessment, what we do is we look at the totality of the data across all these data sources, and we make a clinical decision. So for certain drug event pairs, we may rely more on spontaneous, certain drug event pairs more on observational, but really it's the totality of the evidence that we evaluate whenever we make a determination of a potential causal association. So in the last 7 minutes that I have, I briefly want to introduce going from data, how do we turn the data that I mentioned into information? And really it's done, there's a couple of ways. We do qualitative signal detection and we do quantitative signal detection. Both are done together, but as you can imagine, there's some differences between the two. With qualitative signal detection, we really look at an individual case. I will read every word in that narrative and determine, is that something of concern or not? Generally, we try to focus on, rather than reading every case that comes in, on serious unlisted events that can impact the benefit risk profile, or events of special interest. When these come in, we review these several times a week, and we review every single word, assess it for clinical quality to decide if this is an issue of interest or not. When it comes to assessing the cases, really there's a couple different filters that we use. One, we can, using our medical expertise, we can also look at attributes of the drug event pair. So if I'm looking at anaphylactic reaction and it occurred 4 years after they started the drug, it's probably not the drug causing it. We do look for attributes that would be consistent with the causal association, re-challenge, de-challenge, medical histories, what was your treatment for the adverse event? As well as we use some frameworks as well. The one that probably everyone here knows about is Bradford Hill. But at the end of the day, it's all about data quality when it comes to qualitative signal detection. And then I think this is my last slide. Then moving a little bit into the quantitative signal detection piece. I know that Nicholas and Judy will dive into this more with their talks, but we also have, because we have so much data, we have hundreds of thousands of reports coming in. We also want to make sure we don't miss something. So we also take a quantitative approach as well. There's many different ways that you can look at the quantitative features. And really what we do is we look for either absolute numbers. So when we do a cumulative line listing, if most of the numbers are 1 and 2, and I see a count of 37, well, why is there so many more of this one event? So it could be as simple as some numbers bigger than the others. But then we start getting into more analytics where we start doing disproportionality testing, where we're trying to look for events that are reported at a higher than expected frequency or rate. So there's lots of different methods that can be used, and there's lots of papers comparing these, and some of these methods will be discussed in a little bit. We also can get into some, and really disproportionality broken down into a few buckets. General disproportionality, where we do it for all drug event pairs. There's specialized, if you will, disproportionality, where we focus on a specific subgroup. So we may compare a rate in the elderly versus the rate in adults. We may compare the rate in men versus women. So we may do some specialized screening or some subset screening as well. And then, of course, because this is quantitative, there is a lot of discussions around, well, what is the threshold? And generally speaking, from my experience, the threshold you decide really is a sensitivity specificity trade-off. The higher the threshold, the greater the specificity, but lower the sensitivity, and vice versa for lower thresholds. There was a paper published, it was another IMI Web Radar paper that really looked at, the secret sauce to a lot of the quantitative piece isn't necessarily the method, but it's the threshold that really determines the performance characteristics. And of course, not to mention, there's lots of statistical alerts many of them, I would say the vast majority of them, were not just because there's an increase in relative reporting. Correlation does not mean causation. There's a lot of reasons why you could have increase in reporting that may be related to other than a causal association. There are some advanced methods being explored, whether it be, weighting cases differently, some of the great work the WHO has done, machine learning, artificial intelligence. There's lots of things there. And of course, once we identify a signal, then that starts if you think back to several slides ago where I talked about the signal management process, it all starts with a signal. Then we validate the signal, and then we do the signal assessment. And that's where we determine a potential causal association. Any signal that we start with through a quantitative method still goes through that process and ultimately is reviewed within the context not only of statistics, but also within clinical context as well. And then a decision is made by the scientist-physician pair about a potential causal association. So in conclusion, pharmacovigilance is an emerging scientific discipline. There's still lots of innovation moving forward. There's lots of innovation done over the last 10 years, and there's even more going to be done in the next 10 years, which really excites me, some of the really neat work that Sentinel and others are doing. Pharmacovigilance uses many different data streams. Each data stream has its own unique set of strengths and weaknesses, and really it's drug event pair specific. If I'm evaluating a specific event, generally speaking, one or two data sources are going to be better than the others. It really just depends on the characteristics of the drug event pair. And then as much as people love to complain about spontaneous adverse event reports, and if you search online, there's lots of people talking about the poor quality, yada, yada. It still is our number one source outside of regulatory agencies, number one source of signals in the post-marketing setting. And it really is still the gold standard for what we do in post-marketing safety surveillance. So with that, I think I finished just under a minute early. So I guess I'll turn it back over, or I guess I get any clarifying questions before I turn it back over to Judy.
Hi, Greg. It looks like you have a question in the chat. I'll read it to you in case you can't read it. It comes from Benjamin. He asks, how much overlap do you expect between spontaneous reporting and what you see in the published literature? And is there any way of assessing this?
So that's a really great point, because interesting enough, if we identify a literature report that meets our definition of an adverse event, that gets entered into our database, which then ends up in our quantitative signal detection piece. So it actually feeds into the potential over-reporting. But at a higher level, generally speaking, I don't see much overlap. 20, I can't remember how many, 20-something thousand MEDRA PTs, 99% of those aren't, to be honest, the type of things that you would publish on. Publishing that your over-the-counter medication causes a rash isn't the most exciting thing in the world. So usually what you see in the published literature are those potential game changers, medically serious, Steven Johnson's, and agranulocytosis. It's those big hitters where, there could be some evidence of causal association, but there's actually very little overlap in my experience between those. But in the final point, when we do the evaluation, when we do the signal assessment, we still consider all data sources. So literature still has the ability to contribute to the evaluation. But more times than not, it's actually a very minor data source as compared to spontaneous and observational. So I hope that helps.
Thanks,
Any
Greg.
other questions?
That's it for the right now. And thank you, Benjamin, for the question. Why don't we go ahead and turn it over to Nicholas to go ahead and start his presentation. Again, I'll ask him to introduce himself. And then after that, we'll take our short break before the second half. Thanks, Nicholas. Thanks, Greg.
Sorry, can you hear me now?
We can.
Brilliant. Let me see if I can get my slides back up. Okay, great. So thank you so much. It's great to be here and be part of this this session. My name is Niklas Norén, and I'm the Chief Science Officer of the Uppsala Monitoring Center. So we are a Swedish organization based in Uppsala, Sweden, but we are also the WHO Collaborating Center for International Drug Monitoring. So we work with different aspects of pharmacovigilance, supporting the WHO Global Program for International Drug Monitoring. We maintain and administer the global database of individual case reports, VIGIBase, and I personally head up the scientific development part of the organization. So we focus primarily on advancing the science of pharmacovigilance, working a lot with spontaneous reports, of course, but also done work on observational data and social media, like we heard about in the previous presentation. So today, I'm going to focus on the place for statistical methods in pharmacovigilance and some of its pitfalls. So as was clear from Greg's presentation, I think with the masses of information we usually see in these systems, statistical methods do have an important place and a role to play in our operations. But they come into use in a different way than they would typically do in epidemiology. So sometimes even when the methods are quite similar to those that we might use in epidemiology, the way we apply them are different, because our focus in pharmacovigilance is really on a data-driven approach to discover that which we did not know to look for. So whereas in epidemiology, we typically start from a hypothesis, there's something we want to investigate further, and we can customize our analysis to that specific question. In pharmacovigilance, we start without knowing what might be of interest, and we try to let data lead the way. And so pattern discovery comes into that quite naturally. Statistical pattern discovery is an approach where we look for deviations in data that might be of interest for further scrutiny. So we typically don't start with a pre-specified hypothesis. We may have a scope to say that we're looking in this kind of data, we're looking at these drugs, for example. We try to keep it as open-ended as possible. We want to maintain parallel perspectives on data because we don't know exactly where we will see something of interest. And this also means keeping many different dimensions in view, but also many different data points typically, and also different kinds of patterns. So we might be looking at sort of marginal effects of an association between one drug and one adverse event, but we could also be looking for, say, patterns of higher order interactions, say drug-drug interactions, where maybe two drugs together increase the risk disproportionately for a specific adverse event. But this also means that we must critically assess the outcome of these analyses very carefully at the back end of our analysis, because we're not able to customize and target the analysis up front. We have to bear this in mind and look at these results more critically than we normally would if we'd thought very carefully about what we're going to study and set the study up to address that specific question up front. So the aim of this is usually to be able to identify the unexpected, that safety signal that maybe wasn't on the risk management plan, but something that is occurring in the population unexpectedly, something we didn't see in the animal experiments or in the clinical trials, etc. It can also be a very important way to broaden our perspective. So maybe we see something in the data, but there's something about that, whereas if we sort of let data lead the way and look at other covariates, we might see something that suggests that this has another explanation. It may not be the drug causing this in these patients, but there's something about the nature of this data that could alter the interpretation of this primary analysis. And that becomes then a very important part of our sanity checks for the analyses. So of course, in the broad surveillance context of pharmacovigilance, this is fundamental. This is what we set out to do. It's also very important to include this mindset when we assess the signals, like we saw before. So if we see something that's of interest, could be coming from a quantitative screening, could be coming from a qualitative screening, there may be aspects around that case series that we should be alerted to, and data-driven statistical pattern discovery can help in that context as well. It's also potentially very useful, this exercise, for data management purposes, because sometimes we'll surface something that doesn't really relate to the intrinsic effect of the drug or the adverse event or even of the population we're studying, but it's something about how we've registered data. So for example, maybe we see higher reporting of a specific adverse event than a specific drug in a specific country, and it's really only that country. That could be maybe there's a specific risk factor in that country, but it could also be that there's something in how this data is being coded somewhere in that pipeline, receiving those reports, coding them, sharing them with the international community, that's led to an issue. And maybe that's something that would come out and we could then address and improve the data. So it becomes a big part and an opportunity to get better data to work with further in the future. And that should not be underestimated. So there are a number of different methods available and in use. I think, like Greg talked about in the previous presentation, a fundamental question is, if we see a number, if we're looking at data and we see there are 6 reports on a specific adverse event with a specific drug, what do we make of that? If we're going to try to automate the statistical pattern discovery and help focus the attention of our clinicians on specific case areas, we need to know if 6 reports is something to worry about. And of course that varies. If we have a common event like rash and a common drug like paracetamol, 6 reports would be probably less than we would expect in a big database. If on the other hand, we're looking at acute renal failure, which is rarer, and we're looking at a newer drug, say dronedarone, 6 reports could be something that's worth considering and worth assessing in greater detail. But of course, to automate this and apply this on a broad scale, we need to have a way to know that. And the problem is we don't have access to, especially in the international data, but we don't have access to, we don't know, the number of exposed patients, how many patients took this drug. We don't know how common different adverse events are in the population. We are getting this kind of data now for specific adverse events of special interest in the context of vaccine surveillance, but broadly speaking, we don't have that information available. And even when we do, we know that every adverse event is not going to be reported, and that degree of underreporting varies between drugs, it varies between adverse events, and so that has a differential effect that's hard to factor in. So what has become the general approach to handling this is a paradigm of disproportionality analysis. And the concept was proposed already in, I think, the late 60s, early 1970s by the great thinkers of those times, where they said, well, maybe we can use the database of spontaneous reports as a reference in understanding or getting a proxy for what we should expect in terms of the reporting for a specific drug in an adverse event. And this was then sort of operationalized and put into use on a broad scale by bigger organizations that WHO, the FDA, big pharmaceutical companies, the MHRA were early as well in the UK. But this happened in the late 90s. And since then it spread, and I think it's now fairly commonly used by many of the large organizations that have enough sort of incoming reports that they cannot assess every one of them singly, or even if they do, they may risk missing important patterns because they can't really have the oversight of that entire database. So it's a fun and quirky word, but it basically means not the same proportion. And in this case, we're looking at proportional reports on that ADR. Conceptually, it may look like something of what we see in this graph. So if we think of just one adverse drug reaction, say acute renal failure, and then we look at the different drugs, and so every bar in this graph would represent a specific drug, and its height would correspond to the proportion of it, the total number of reports we have on that drug that actually relate to acute renal failure. And so the basic assumption is that we have some sort of background rate, the background relative reporting rate that represents how often this occurs in the population and is being reported. And then we're looking for deviations like we see in the rightmost column that could represent dronedarone. And then we see that we have an observed relative reporting rate, which is vastly exaggerated here, but that would then deviate from that expected proportion. And this is the fundamental notion of disproportionality analysis, to look for such deviations and see which ones may be worth looking more closely at. In practice, this brings us to a simple 2x2 table. So if you just think of one drug and one adverse event, we can of course sort of characterize the data in just the sense of those two, like we would in normal epidemiological methods as well. So we get a count A that represents the number of reports with both the drug and the adverse event, so the size of the case series. We then have B, the number of reports with the drug, but not the event. C, correspondingly, the number of reports with the event, but not the drug. And then D, typically the biggest number, because we look at rare drugs and rare adverse events, that would be the number of reports with neither the drug nor the adverse event. So based on that contingency table, we can then look at the observed and expected counts. We typically shift focus and not look at the proportions, but we look at, well, how many reports do we have and how many reports would we expect? And like we heard in the previous presentation, there are a variety of different measures of disproportionality. I'm going to show you in detail just one, but I'm going to talk a bit about differences between them on the following slides. But if we look at the simplest one, which I think is fair to say, it would be the relative reporting ratio. And basically, like in all of these cases, the observed number of reports is always A, that count we see there. And then basically, when we compute the relative reporting ratio, we get an expected count by taking the total number of reports on the drug, so A plus B, and we multiply that by the overall proportion of reports in the database with that adverse event. So A plus C divided by the size on the entire database, so all the counts in that contingency table. So with that observed count and that expected count, we can form an observed to expected ratio that we can then look at to see, is this worth looking more at? Now, in that case, you may have noted we included the drug of interest in the background rate. So when we computed that expected rate, we took the entire database, which means that if we're looking at dronedarone is also part of that background. And of course, in terms of contrast, it's a less sharp contrast. So conceptually, it may be better to sort of take it out. And if you do, you get what's called a proportional reporting ratio, which is also very broadly used. Now, both of those look at these rates of reporting only, but you could, of course, also look at an odds ratio instead and assume that the odds are constant in the different under the different treatments, and then based on that odds ratio, get to an observed to expected ratio as well. And of course, mathematically, that is stronger. It's a better measure in the sense that it's variation independent under the marginals and has some other nice properties. In practice, though, because we're looking at rare events, as long as we are looking at rare events, it doesn't really make much of a difference. These are all in the same direction. So if you have a positive odds ratio, you'll have a positive PRR, you'll have a positive RR, and so forth. So I don't think, and I think this echoes what Greg said, it's not the most important choice, but it is a choice you have to make. But you can rest assured that for the most part, they're only going to differ very slightly between one another as long as you look at the rare events. The other big question we have, of course, though, is how do we handle random variability? Because these measures could become quite large even when we have quite little data available. So if you're looking at a very small case here, so just 1 or 2 reports, you can have a very low expected count in a big database that would, throw that out and make that quite big. But it still wouldn't be most likely something you want to look at because you just have 1 or 2 reports. It's unlikely to maybe represent something worthy of further consideration. So of course, you can then start to think about statistical significance. And among the first approaches that were proposed to address this was composite criteria. And this is an example from Stephen Evans and colleagues in PDS, where they said, well, we want to look at the proportional reporting ratio. We only want to look at combinations, cases where that exceeds 2. But we also want to look at statistical significance, of course, to make sure we're not looking at stuff that is likely to be representing random variability or noise. So we're going to do a statistical significance test. I think they used the chi-squared test in their case and put a threshold on that. But it turned out that wasn't enough to protect you against those very small cases when you have very low expected counts. So they also added a third criteria saying that we're not going to look at anything unless we have 3 reports. So you end up with these composite criteria like this. And there are a variety of those. And the IMI study that Greg talked about earlier actually looked at a number of different ones from different regulatory authorities and also different pharmaceutical companies. Another approach, if you want to look at statistical significance, and this is a more recent proposal by Wang et al. From the FDA, is to instead do a likelihood ratio test. This is based on the same observed and expected counts as we have in the relative reporting ratio, but they use Monte Carlo simulation to be able to set the threshold for alert in a way that you control the false discovery rate. So the false discovery rate is basically, well, what proportion of highlighted associations will be real as opposed to like being generated by random variability. It is, of course, important to remember that this will not factor in bias, which is typically more important than random variability, perhaps, in this data. So when we say we have a false discovery rate that's controlled, that's from noise and not from systematic variability. The other sort of paradigm, and this has actually been around a long time, these were the first approaches that were published for disproportionality, is to use what's called statistical shrinkage. And you can use a Bayesian approach or an empirical Bayes approach, and there are a variety of them that you can do. I'm going to show you the simplest one here. But the idea is that if we take an observed to expected ratio like this, where we have 2 reports and we expect 0.01, obviously that becomes 200, a big number, so it's a big observed to expected ratio, even though it's probably driven by that low expected count, and that could be an artificially low expected count. So it's likely not to be something we want to be looking at. So statistical shrinkage has different mathematical foundations. In the Bayesian context, you may assume that you have a prior distribution, assuming no association between the drug and the adverse event in the absence of data, and then as you get data, you moderate that estimate. But that prior assumption will pull your estimate towards the baseline assumption of no association, which is an observed to expected ratio of 1. In practice, with the simple shrinkage I'm going to show you here, basically what we do technically, this is mathematically a Poisson gamma model, but technically it just means we add 0.5 to the denominator. So it's like assuming there's an artificial extra half report, but we also add the same amount to the expected value. And you can see that if you have a small observed or expected count like we do now, you actually have a quite large effect because now we divide by half and not by 0.01. So we pull this down to 5. So you have quite great effect for small case series, but if you have a bigger case series, if we make a comparison here, if we think about 2 versus 0.01, like we saw there, we look at 20 versus 0.1. So we just multiply them by 10. They're the same observed to expected ratio. So this is before shrinkage. But when we add the shrinkage, we see that the observed to expected ratio based on just 2 counts is much more effective than the other one. It also has a broader confidence interval, credibility interval, because there's more uncertainty. And combined, it means that we wouldn't highlight that particular association as a statistical association if we use this statistical shrinkage with credibility intervals. Whereas the other one with 20 reports when we expect 0.1, that is probably interesting because such a big deviation, and we have enough data to be able to trust that it is something that's sort of statistically robust. So just to wrap up this part, and this will echo what we heard before, I think the choice of measure, the choice of approach is not really the most fundamental one you do. I mean, they're all quite similar. I do think the choice of threshold, like we heard before, is important in terms of your balance between false positives and false negatives. But in terms of some of the pitfalls and what I'm going to talk about for the rest of my presentation, they will be equally affected because many of those problems come from the fact that all these measures are just based on this simple contingency table of just simple counts of A, B, C, D, where we just look at one drug and one ADR at a time. And also the fact that we just treat every report equally. So much of, if not everything, what you'll hear next is applying to all of them before. And this is the paper by Kandor et al. Is that paper from the IMI Protect project where they looked at different measures and were able to show that the most important choice is actually the threshold and not so much the measure itself. Okay, I'm going to switch sort of pace a bit there, focus. This is a bit of a crash course into that basic paradigm of disproportionality analysis. And I'm going to show you some of the potential pitfalls. And the first one will be an example where we look at something that turns out not to be interesting in the end. So if we look in the database and we do statistical screening with disproportionality analysis, we might see a pattern like this one where for growth retarded with poliovirus vaccine, we have 17 reports and we only expect 7. So on the surface, this may look interesting. But of course, you're a trained epidemiologist and you are astute and alert to the possibilities of confounding. And here, of course, there should be alarm bells going off because we know poliovirus vaccine is primarily given to infants. and why, you know, we all suffer from growth retarded. It's really only, I think, considered an adverse event once you're an adult. So this should also be reported primarily for the young. And indeed, if we break the data down by age group, we do see that in the infants, where we have the most reports, there's actually clearly less reports than we would expect. And the same is true in the toddlers, where we have 2 reports, we expect 5, and in the other age groups, we don't really have much data at all. So what's happening here is when we don't do this age breakdown, but we actually look at the entire database together, we add in a lot of reports that are neither on growth retarded or on poliovirus vaccine. And by increasing the denominator, the relative reporting rate of each of those grows quite significantly. And even though the database size is also growing, that's not enough to counteract that when we have that effect on both growth retarded and the poliovirus vaccine. If it had only been the one, it would have been fine, but when it's both, we get confounded. So this is an example of Simpson's paradox, where we actually see a consistent pattern in the different subgroups where we have data, but when we put them together, not accounting for that covariate, we reverse the association. This kind of problem is actually not the biggest concern in a way. This is a problem in efficiency, because most likely the clinicians will see this, they'll spot this when they look at the cases, and they'll be able to say, well, dismiss it and say, well, actually, this could still be interesting. But if it was driven just by the fact that we had more reports than we expected, it shouldn't be, because actually there isn't when we account for age as a covariate. And they will be able to tease that apart. A bigger problem, I think, is my next example, where there's something we wouldn't see in the statistical screening that could potentially be interesting. So here we look at alopecia, so loss of hair with fluoxetine, antidepressant. And we have a lot of reports, nearly 300, but we expect more reports than that. So this would not come out of the statistical screening. However, if we now break down data by the different countries, we see that in 3 different countries at least, we have quite significant disproportionalities in the reporting. So in the UK in particular, there's over 100 reports and we expected only 65. In Canada as well, we have 22 reports, only expected about 4. And in Australia, 14 versus 8. So there are more reports in these countries than we would expect, but we're not seeing it. And the reason we're not seeing it is that in the US, where we have a lot of reporting of fluoxetine and of alopecia, we actually have less reports than we expect on the 2 together. So we have 65 reports, but we expect 250. And in this case, it means that because of this heterogeneity in the data, we wouldn't be able to pick this up with adjustment by stratification either, because there is that heterogeneity. When we pool those stratum-specific estimates, this US reverse pattern is enough to cloak the pattern in those Commonwealth countries. So this is something that potentially we should be looking at, because there is an over-reporting. Maybe we want to spend time on it, but it's a problem if you just use these crude analysis, or even in this case, the adjustment by stratification, we wouldn't be seeing it. It wouldn't be brought to our attention. And that is something to be on the lookout for. So what you can do, what you'd have to do to handle this is you have to basically subgroup your data. We proposed a way to do this. The difficulty of doing that, of course, is you get a multiple comparisons problem, because when you subgroup the data, and if you subgroup by different covariates, so if you want to look at both different age groups, you want to look at men and women separately, you want to look at different countries separately, maybe you want to look even sort of different combinations, so maybe you want to look at boys separately from girls, etc. We've not done more than 2 at a time, and usually we just do 1. But even at that, you have to be worried about or mindful about your multiple comparisons, because you could induce false associations, because now you look at every association, not just in one way, but in a multitude of ways. So if you have to handle that in a robust way. The paper by Seabrook et al. Is also out of the IMI Protect project. It compared stratum-specific sort of analysis to adjustment by stratification and to the crude analysis. And they actually showed that in pharmacovigilance, oftentimes subgroup analysis bring more value than do adjustment by stratification, presumably because we actually have quite a lot of heterogeneity in the data. And the adjustment by stratification assumes that you have homogeneity, so it makes sense to pool your estimates. So that they make the case, or they show the benefit of doing it. But I don't think it's something that's in broad use in sort of a data-driven manner, at least. I think several organizations may look at children, but not many organizations yet have figured out how do we do this in a bigger, broader context to be open to more kinds of subgroup patterns that could be of interest. We have used it for some time at the WHO Collaborating Center, and this is one signal that we found when we did it. It's ceftriaxone hepatitis. It was published by Boyd in the WHO Pharmaceuticals newsletter. Again, on the whole, we see less reports than we'd expect on this adverse event with this drug. But in this instance, when we did the subgroup analysis, a pattern for the elderly popped up. So we saw that in the elderly, we had 70 reports nearly, and we expected about 40. And so this was brought to the attention of the clinicians. And there are going to be lots of patterns like this that are not interesting in the end. I mean, it's just be something else, as always, like we heard before. But in this instance, when they looked more closely at it, they became convinced that this is a likely adverse drug reaction, at least something we should communicate more broadly and bring to other people's attention, because the case series as such was quite strong. We had nearly 50 positive D challenges, so patients where the patient stopped the medicine and the hepatitis resolved. And in 50 of the cases, there was a plausible time to onset as well. And beyond that, ceftriaxone is known to cause raised liver enzymes, and there was evidence to suggest that the elimination half-life did increase in patients over 75 years of age. So there was a potential mechanism for that risk factor of age with this adverse drug reaction. Good. All right. Now, another example of a reason why we may miss something we should be looking at, we would want to be looking at, is what's been called masking. It's been called by other people cloaking, and it's also been called, I think, competition bias. So this is a baby with many names. Just with an example of it, if we look at rhabdomyolysis and we look at venlafaxine, we actually have less reports than we expect. So it would not be highlighted with disproportionality analysis. But if you worked with spontaneous reports a bit, you may know that rhabdomyolysis is very heavily reported with the statins because of historical safety signals. And of course, that in fact, they do cause rhabdomyolysis. And in this instance, if we take out the statins from the analysis, we get a quite different picture. So the observed count dropped, so the case series shrunk, but only by 3 reports. So there are only 3 reports of the reports with venlafaxine or rhabdomyolysis that had the statin co-reported. In those cases, it could of course have been the statin. So, but we take them out, we increase the observed count by 45, but the expected count shrunk from 60 to 15, so to almost a fourth of what we saw before. And all of a sudden, we have more reports than we would expect. So what's happening in this case, if we go back to this principles quick sketch from before, is we have this assumed relative reporting rate of rhabdomyolysis based on which we should get the expected count. But with the statins now, not only do we have much higher relative reporting rates, but also we have a high, observed number of cases. So they have a big influence on that overall relative reporting rate. So when we include the statins in computing that overall reporting rate of rhabdomyolysis in the database, it's 4 times as large as if when we exclude them. And of course, excluding them is more, it better fits the original assumptions we made of this sort of background relative reporting rate. Here we have very high reporting rates because they actually cause this adverse drug reaction. And when we inflate that overall reporting rate, we inflate the expected count of every other association with rhabdomyolysis, and correspondingly, we sort of shrink the observed to expected ratios in a way that we don't want to. This is not an even problem. If we look at the most influential drugs on the overall reporting rate of rhabdomyolysis, we see that there are these 4 drugs. They're all statins. And we see that if you look on the x-axis, you see 40% for that rightmost dot. It means that if we take out this drug, this is rovastatin, the overall reporting rate of rhabdomyolysis in the database will drop by 40%. And similarly, there are 2 statins that if we take them out, they'll drop by 20%. And that shows why we have such a big impact here. If we look at some other adverse event, and in particular diarrhea on the far right, we see that there's no drug that has any real impact on that overall reporting rate. So we have a much more even reporting. And this is the case for most of the drugs and most of the adverse drug reactions in the database. In most cases, there is no masking. But when there is, like there is for rhabdomyolysis and the statins, there can be a a substantial effect. So there are ways you can handle this. One is to do a regression. It's been proposed by different authors. This is one paper that we published, where basically you end up conditioning on the different drugs. So you take out that effect of the statins because it's actually included in your model. We've also proposed a simpler approach where we identify outliers and take them out of disproportionality analysis, and that can be easier to interpret. But they each have their pros and cons. I haven't seen them in broad regular use, these kinds of approaches, even though they've been around for some time. So I think this is something you still need to be mindful of, that you could be suffering from depending on what you're looking at. Okay. So one big problem with the statistical methods, and disproportionality analysis in particular, is that it treats every report the same. So it counts every report equally, even though we know all reports aren't equal in terms of the amount of information they have, and maybe what that information conveys in terms of the likelihood that the drug had caused the adverse event in each of them. And of course, in the clinical review, that information is critically important, and the assessors will place very different weights on different reports in their assessment. And this is a big gap in how we do the statistical screening normally, and then how we do the assessment, and then also a big opportunity for improvement. And just to show you what this may look like, another case series we wouldn't find if we just did disproportionality analysis is photosensitivity with Montelukast, 19 reports versus about 30. So we wouldn't pick this up. It's important to note that we'll not be able to pick this up by subgroup analysis or stratification or unmasking. So none of what I've shown you before will make this stand out. It just doesn't have more reports than we'd expect. But what it does have is some quite strong reports. So we have reports from 8 different countries. So there's a geographic spread, which increases maybe our suspicion that this is something real and something true. Montelukast is the solely suspected drug on almost all of these reports, all but one. And then we have 3 patients who stopped Montelukast and the photosensitivity went away, and 2 patients that then restarted Montelukast and had the photosensitivity come back. So very strong cases, those 2. So this became a signal at the time, but it just illustrates that there are going to be these signals that are of interest, and they won't be driven by having more reports than expected. So is there a way we could bring these case series to the forth and to the review in a systematic and reliable way with a computerized method? So there have been a number of initiatives to try this recently. The first 3 papers here are all methods for case series, which try to build predictive models of scores for case series based on multiple aspects of strength of evidence. So in the Custer et al. Paper, we considered geographic spread, for example, the amount of information on the reports, still disproportionality analysis. This is still part of it. There are also a number of other covariates that didn't become part of the model in the end. Similarly, Scholl et al. Also looked sort of more broadly at different aspects around the case series. Maybe was it reported by a consumer or by a health professional? Did you have a positive challenge, etc. Van Holle and Beauchot combined disproportionality analysis with time to onset analysis. So that's also an interesting approach, but it still used regression, all those 3 methods. The paper by Munoz et al. from FDA is different in the sense that it actually is also looking at multiple aspects of strength of evidence, but it's trying to identify strong case reports for review, which is also, of course, a very interesting and important challenge for the future. So I think we'll see more of these in the times to come, because I think there is really a lot of strength and opportunity in looking more broadly than just the numbers of reports and going beyond that. Okay, now the final challenge and limitation and pitfall I'm going to talk about is that of just considering one adverse event term at a time. So most of the analysis we do, and what I've shown you now, is we look at one of the adverse events that tend to be reported on a specific case. And that can give a quite fragmented and incomplete picture, not only because, adverse events can be reported differently, but also because a single adverse event may not convey the full clinical experience of a specific patient. So if we consider a simple example like this, where 4 reports of possibly similar conditions. This could be a sort of a spectrum of cataplexy or narcolepsy, for example. But each of the the four reports look quite differently. So they don't share any single term. And if we take one of these terms, say cataplexy, we would only get the top left patient. We'd miss the other three. If we go for something broader like fatigue, we would get the two patients on the right, but we get a lot of other reports that are not related or not relevant to this case series. So we have this problem of fragmentation because coding is different, clinical presentation is different. And to some extent, we can address that if we go up in the hierarchy and say MedDRA and we look at a higher level term. If we're able to catch all the relevant terms, that's not always the case, but say we do, there can still be the problem that in a patient, it may not be enough to say they had fatigue, but maybe fatigue together with somnolence and depression mean so they meet our criteria for inclusion, and that's not easily done. So if we don't find ways to account for this, we will be at risk of both missing important cases, so fragmenting the case series, but also diluting case series by including cases in the case series that really don't belong there because they don't really meet they represent another clinical condition that just shares a sign and symptom with what we're interested in, for example. So one method we've been working with for quite a long time, and in particular in the most recent years in view of COVID vaccine surveillance, is adverse event cluster analysis. So the idea here is to identify clinically coherent groups of reports, not by just looking at single adverse event terms, but by using unsupervised learning, considering all the reported adverse events on each report and try to see which ones are most similar in a data-driven manner. So the methods paper, we're still in the process of publishing, but here is a paper we published where we use that method to look at a historical safety signal in drug safety. Another approach that we've been looking at is ways to, and this is not looking at every adverse event on each report, but smart ways to maybe link adverse event terms together that may describe the same clinical condition, but may not belong in the same part of the hierarchies, is being adopted from distributional semantics. And this comes from the analysis of free text and natural language processing. And the idea here is to use these deep neural networks and build a predictor for adverse events reported or included on a report, given the other drugs and the other adverse events that are reported there. So by building that predictive model and trying to predict something being left out, we can learn something about the weights for each of the adverse events. They actually become a representation, a vector for that adverse event. And ideally, and this has turned out to work quite well in practice, if those vectors are similar, they tend to be adverse events that are used in the same context. So they may not be reported together. It could be acute renal failure and renal failure, which typically wouldn't be on the same report, but they might be in the same context. And this type of an approach allows us to find those patterns and then link those adverse events and bring them together as a help in, for example, building our case series in the signal assessment. So this is also, I think, for us, it's at the forefront now. It's something we're focusing on for the future. And I think it's something we'll see coming also in the near future. That is essentially what I wanted to share. I'm just going to note in the end that I do think statistical pattern discovery can be an excellent servant. I think it is necessary and a great help for many of us that work with large data sets. But we need to remind ourselves and be mindful of all these limitations and potential pitfalls and proceed with care so we don't step into one of them. Great. I will end there and see if there have been any questions or if there are any questions of clarification at this stage.
Nicholas, I'm monitoring the chat and I don't see any questions, but I want to invite folks, if you have any clarifying questions or specific questions to Nicholas's presentation right now. We're running about 5 minutes ahead, so we have time for a few questions. And then if none, we can go ahead and proceed to our break. But I want to give the audience a chance, if you do have any questions, to go ahead and ask them now. All right, folks, seeing none that have popped up, we have about a 10-minute break now. So why don't we go ahead and resume at 9:40, get some coffee or water. I'm going to go ahead and put up the link one more time to the course materials so that if folks have joined our session midway, that they have access to this link. A reminder that our next session is going to include a live interactive exercise. And if you'd like to be part of the exercise, it's important to download these materials and have it available, hopefully on your machine, so that you can walk through the exercise with us. And if folks don't have that, that's okay too. We're going to walk through it whether or not you're going to follow along live. So either way, we'll see you again at 9:40. We are ready to go ahead and get started again. Thank you for folks. We know that's a really long morning. Thank you for sticking with us through all the presentations today. Really appreciate it. I'm going to stop monitoring the chat and ask my fellow panelists to monitor on my behalf if there are any questions that come up. Briefly, my name's Judy. I am an assistant professor at the Department of Population Medicine, which is located at Harvard Medical School and Harvard Pilgrim Healthcare Institute in Boston. I have no conflicts of interest to disclose. I am also privileged that the Harvard Pilgrim Healthcare Institute houses the FDA's Sentinel Operations Center. And as a result, a lot of the material that you'll see today was developed over many years with respect to methods development in the signal detection and signal identification space. And so I hope you'll see a lot of connections with some of the discussions we've had earlier today with Greg and Nicholas. I'll try to point out where those are as we go along. And thanks for being here. So what are we going to talk about today? We're going to look at signal identification, particularly in longitudinal databases, which Greg touched on earlier as the observational data component. We're going to go ahead and do a demonstration of how we would use a method like this. A tree scan method is what it's called, or a log likelihood ratio method. And we're going to do this with a simulated data set, and we're going to assume a propensity score match design. And then we're going to go through, as we do this demonstration, a live exercise where if you guys would like to, you can do your own signal detection on your machines if you've downloaded the appropriate software. And so really briefly, to sort of bring some more material back to some of the things Nicholas talked about in the last presentation, trying to look at where there are similarities and where there are differences between signal identification as performed in spontaneous reports versus signal identification as performed in longitudinal databases. So in both cases, the idea is the same from the outset, which is that we are not in the business here of looking at a particular exposure outcome pairing and then trying to do an evaluation of that outcome pairing. Instead, we start almost from a blank slate where we say, okay, we have this one particular exposure of interest and we're going to look at all comers in terms of outcomes. So same idea there as spontaneous reports. Both of them then are really geared around hypothesis generation, with the idea that any hypotheses that have been produced will then necessitate further investigation and most likely a formal follow-up study of some sort. Although Greg did point out that some of the formal follow-up ends up being case reports and causal analysis. And sometimes the signal potentially so strong that it's not necessary to go to a full pharmacoepi study. But the idea here being is that when you do this signal identification, you're not done sort of as soon as you find a statistical signal. You have to go through some follow-up, and the idea is this is the beginning of a journey rather than the end or the conclusion. In both cases, the spontaneous reporting data and also the data that we use here in observational databases can be structured into hierarchical trees to build power. And so this touches on the slide that Nicholas just finished on in his last presentation, where there are potentially in spontaneous reporting systems, there's the ability to go to what are called higher level terms. So basically look for bigger clinical groupings of things to try to understand what signal might be present. Just like there's a hierarchical tree in MedDRA terms, which I'll show an example of in the next slide, there also is a hierarchical tree in diagnosis codes that are commonly used in observational data. So the ICD-9 system and then the ICD-10 system that followed it is arranged in a hierarchical structure. And so one of the ways that you can leverage that structure is to look at these higher level clinical groupings to be able to find signals that potentially wouldn't be available if you were looking at just one particular code. And so some of the differences, they obviously start with different data sources. So longitudinal data sources are more compatible with familiar epi designs because they're the foundation of a lot of them. And those epi designs in the past have often analyzed particular exposure outcome pairings. They have different analytic data sets. So longitudinal data can be analyzed as a summary level data set, an aggregate data set, whereas often, as we just saw in some of the discussions that that Nicholas and Greg presented, is you're looking at in a spontaneous reporting data set, a case report. And so there's really deep potential patient level information in the sort of most valuable reports, back to the animal farm idea. So that is not something that we see here in observational data typically. There is differences in multiplicity control. So some of the methods and the ones that I'll present today are going to have very explicit multiple hypothesis testing control. Others use different types of methods. Shrinkage is an example of a method where it's intending to, well, shrinkage is actually, it's a different, let me back up and say shrinkage is used for a different reason, but there are other things that can be used to try to prevent problems that occur from multiple hypothesis testing. There are different comparison groups. So here in the previous couple of talks that you've seen, the idea was to compare the reports of a particular exposure outcome pairing to all other reports. And so with a longitudinal database, you can further specify a very specific comparison group. And in this case, what we'll talk about today is a propensity score design where we're comparing drug A to drug B, which is atypical of what you would do with a spontaneous reporting database. And so, as I mentioned, just sort of an illustration of how these terms are grouped together, or these clinical outcomes are grouped. On the right here, you see an example with MEDRA terminology. So typically, when spontaneous reports are utilized, the preferred term tends to be the term that's most commonly used. But as Nicholas mentioned, you can go to a higher level term if you want to build more strength in terms of more reports that then factor into your analysis. Similarly, at the lowest level of a tree, and in this case, I'm showing an ICD-9 tree because that's going to be what we use in our toy exercise today. There are individual diagnosis codes that then group up into an aggregated clinical concept and then further group up as you go up the tree. And the idea here, which I'll talk about a little bit specifically, is that what the value of this is that you don't have to have clinicians all coding identically. There may be some clinicians where the practice is, for example, in their particular healthcare organization to code convulsions this way. So with a pretty general code, and that's what people normally use. Other practices might code this way. And the idea is if it's really very similar in concept, that we'd like to build strength and use that combined clinical grouping to be able to potentially find any signal of interest. And so just trying to tie all of these things together, there's been kind of a convergence on looking at these different statistical techniques with these different data sources. And so as we just learned, spontaneous reporting systems primarily use disproportionality analyses, and those can come in frequentist flavors, and I'm showing you a couple of those there, or they can come in the Bayesian style, which was the shrinkage example that Nicholas went through. And so all of those at base are setting up this observed versus expected scenario, which Nicholas showed with the contingency table. And so typically the comparison, though, is to all reports that are available on all drugs and outcomes. And then there's potentially the stratifications, or as Nicholas pointed out, also the subgroups are another way to go about analyzing that data. So likelihood ratio test methods, which were touched upon just briefly in the paper by Huang et al. That was referenced during the last talk, those also characterize observed versus expected counts. But one of the main differences here is even when applied to spontaneous report data, there's that explicit control for multiple hypothesis testing, and it's done through that Monte Carlo simulation component. And so recently there's been a lot of comparisons between a tree scan like or a likelihood ratio test method and traditional disproportionality analyses. And multiple comparisons in both the empirical and the simulated data have similar findings. You can see some of the references I put on the screen here. Essentially, the tree scan and the log likelihood ratio methods in these papers are shown to have better false positive control, whereas the other methods there are shown to have more false positives, but also fewer false negatives or missed signals. And one of the important points of this is when these are tested in these particular papers, all of them, as we talked about before, rely on these tuning parameters, which allow you to have different implications for your type 1 or type 2 errors. So I'm calling them tuning parameters, but earlier, Nicholas and Greg referred to them as thresholds, but it's the same idea. It's essentially saying that, if I keep my threshold really low, then I'm going to potentially allow a lot more in the door to examine. If I set my threshold really high, then I'm going to allow a lot less in, but then I might miss something that isn't quite at the level of strength that's needed to sort of meet my threshold. And so in general, the idea here is, again, all of these are sort of geared around taking data, formatting it into that same contingency table and trying to generate some understanding from that data, even if the underlying data source is spontaneous versus longitudinal. And so here is just a little diagram of some of the types of methods that are available using longitudinal data. So I start on the left with the idea of the study design. And I think it's really important when you're using longitudinal data, because you have the availability of the denominator and that availability to follow people over 3 or 5 or 10 years, in some of these European databases that are super rich in terms of longitudinality. To make sure that you set up from the start about what epi design you might be using to think about how to analyze your data. And so a common design that's used is a self-controlled design, and that has a couple of techniques that have been used in the literature to look at it. So one are the likelihood ratio test methods, which includes the tree scan based analytics. One is sequence symmetry analysis, and one is information component temporal pattern discovery, which has been operationalized into a tool that's called a chronograph for those who are familiar with it. So that's one type of design. We're not going to spend a lot of time on that today because it's not part of our exercise, but I included some slides in the course materials that you can look at your leisure that include some data about self-controlled designs. What we're going to spend time on today is the second category, the propensity score or the fixed ratio match design. And so in this case, we are choosing actively a comparator group. So we're saying, okay, we're interested in monitoring folks who are exposed to drug A, and we're going to then choose to compare them to what we think are similarly situated folks exposed to drug B. So the idea here between the choice of drug A or drug B is that drug B is typically going to be something that is a reasonable substitute for drug A. Maybe it's another drug in class, maybe you cross classes, maybe it's a group of all drugs in class. But the idea here is that unlike what's typically available in spontaneous reports, you're going to choose to actually ask a different research question, a different question with respect to that active comparator. Finally, another type of design is a stratified cohort design. This is very similar to the propensity score based, because often the stratification is based on n tiles of the propensity score, although it could also just be much simpler. It could just be sex and age, for example, but they're all in that cohort design. And so what we're going to talk about today is the likelihood ratio methods associated with those cohort based designs. And so some of the work, just so you guys are familiar, if you feel like taking a look at it, associated with these propensity scores and signal identification starts here in 2018. And so this is taking a look at setting up a propensity score match cohort and then looking at that cohort for, again, all outcomes using this likelihood ratio based or tree scan based analysis. So one of the things that I think is important to point out, and that maybe throws people a little bit with propensity scores, is typically with your propensity score, you might choose the covariates in your propensity score that are in fact related to both your exposure and your outcome, or those that are related to your outcome specifically. And one of the challenges of bringing a propensity score paradigm into a signal identification paradigm is now you're not looking for one particular outcome. you're looking for 8,000 outcomes. And so the calculus of what goes into your propensity score is slightly different. And so we spent some time building what we think of as a general propensity score for signal identification. So the idea here is that the components of the propensity score are going to be general risk factors for a wide variety of outcomes. And so they include things like frailty, they include things like concomitant medications, things like that, that we think of as general purpose risk factors, as opposed to being covariates that are specifically related to an outcome of interest. And so this is just one last paper that just came out that I put here just to show the use of this technique in common longitudinal databases. And so now we're going to go to the part of the talk where we're going to look particularly at how we would use one of these tools and to go through the exercise with a toy data set that we've created for this purpose today. And so again, we're going to be looking at tree scan, which is a log likelihood ratio based method. It is signal detection or data mining is sort of this category. It leverages data that has been grouped into these hierarchical tree structures, hence the name tree in the name of the analysis. And it automatically adjusts for multiple hypothesis testing using that Monte Carlo style simulation mechanism. And here is the website for the software. And if you're going to follow along with us today, I hope you've downloaded it. And so some of the things that are the strengths of this type of a log likelihood ratio method are that, one, it takes advantage of the hierarchical nature of the clinical concepts, which we talked a lot about earlier in this talk. And part of that is that the investigator doesn't have to know the particular coding practice in any particular healthcare organization. So the idea is that that tree allows you some freedom of not having to understand where the coding is actually happening or the coding practice. And that's especially true when we're looking at many, many outcomes. And then of course, there's the formal control for multiple hypothesis testing, which preserves an overall type 1 error. So some of the limitations, very similar actually to a limitation that Nicholas just pointed out in his last part of his talk, is that all outcomes are treated identically across the tree, regardless of their time of onset, severity, et cetera. So when basically the program is looking at counts, one case of Stevens-Johnson syndrome is just the same as one case of cough. So there's no sort of hierarchy associated with treating one outcome as being more important than another, et cetera. Similar to how Nicholas pointed out that, with the spontaneous reports, all the reports are treated equal, regardless of the quality of any individual report. One of the other components too is that we're looking at the individual occurrence of an outcome and counting that, rather than sort of a more complex definition of outcomes. So often in pharmacoepi studies, in order to increase the positive predictive value of a particular clinical concept, you might say, well, I need 2 outcomes to occur within a certain number of days of each other to believe that this is actually an outcome in administrative claims data, for example. We don't have that level of complexity in something like this type of a design, where the idea here is signal identification and broad scanning, because we don't sort of personalize each of those outcomes and set them up differently. We're just doing one sort of plan for all of the outcomes in a tree, which can be several thousand, and in the ICD-10 era is in the hundreds of thousands actually. And so the other important limitation is that these types of longitudinal analyses start from a specific study design that I showed in the beginning. So am I doing a self-controlled design? Am I doing a cohort design? And the limitations of those study designs get carried forward into the analysis. And so any of the concerns that one might have generally with some type of a study design are not alleviated, for example, by doing a signal identification analysis versus a signal evaluation analysis. And so this is a display of what that software looks like. Just broadly, if you've downloaded this to your computer, which runs on any of the standard operating systems and has a user guide, it's got these 3 tabs, which we'll go through today. It's got the analysis tab, the input file tab, and the output file tab. And we'll walk through how to use this in order to do your own analysis. So any observational design that yields an input data set can be used here. And so the two styles of statistical analysis here are what's a Poisson model and a Bernoulli model. And those are used in different circumstances. For today, our actual incoming data set is here, ICPE 2021 training, and it's got this special ending, which I mentioned before. It's just a text file, but TreeScan understands it better if it has this special ending to it. So that's why it has the special ending. And the tree structure for the data is this. I noticed that in what I uploaded was the text file instead of the TRE. Either way, it will work for today, but just to understand. So basically, these types of sort of data sets are needed in order to be able to run the program effectively. So again, sort of the components of this are the underlying analytic data set, and then how to arrange the outcomes in a particular tree that's utilized. So again, back to the idea of the different study designs going with different types of data. In a Poisson-based likelihood model, you're basically counting a set of observed outcomes and you're comparing them to the expected, but here the expected is based on a referent rate. So you're choosing a referent population, which can be, again, a comparator drug or something else, and you're using that referent population to derive an expected rate. And that rate becomes deterministic in a Poisson-based design. So we often think of that as a one group monitoring scenario because our referent rates can come from historical data or concurrent data, but really the main driver of a Poisson-based model is to use the observed outcomes in your treatment group. Whereas in a Bernoulli type of analysis, you're doing a two group analysis, and you either can do a self-controlled risk interval design where you're using data for the same person in a period of time you designate as the risk window and a period of time you designate as the control window, and then comparing across that person. You also could do it with a case crossover design, which is also another version of a self-controlled design. In this case, although you index on an outcome, you still have these two periods of time within the same person you're comparing. And then finally, there's a fixed ratio match design. And again, for us, that's what we're going to do today. We're going to have a treatment group. We're going to have a comparator group. It's going to be a cohort design. For the way that we get to this fixed ratio match, you can use the propensity score, which is what we'll talk about today. But you could also use just exact matching. You can match on sex and age or some other characteristics that are important to you. And so this is a design diagram of how you would gather your index cohort for the propensity score design for the purpose of doing signal identification. So it's a little bit more complex than what we might normally think of as being involved in a signal identification exercise, but the whole idea here is for the design to make the two groups comparable. So the observed to the expected ratio is more meaningful than it might otherwise be. And so we start out here and we look for both our treatment group and our comparator group, our two different cohorts, to have an index dispensing or administration of a particular drug. We typically then look for that to be a new index dispensing or administration. And so we actually require that we have a period of time before where they're not, have no evidence of having that particular dispensing or administration. And during that time, we typically require enrollment to ensure we have complete capture of that person's history. We may have other inclusion or exclusion criteria that factor into our analysis. For example, we might exclude pregnant women because we decide that this analysis is going to be only about non-pregnant persons. We might exclude children because we might decide that what we're doing for our analysis here is only going to be certain age groups. And then we're going to measure covariates that feed into our propensity score over the same window for which we have enrollment. And so, as I mentioned before, in the toy study design we're talking about today, we would use something like that general propensity score model that I showed that was developed in a prior paper. So we might have information about frailty. We might have information about healthcare utilization during this time. Essentially, the idea here is, again, to create these comparable cohorts so that we know that we're, hopefully comparing apples to apples. And then there's a couple of other pieces of information that we might ensure. We might say, for example, we're only going to follow people once. We're not going to allow them to exit the cohort and re-enter the cohort, et cetera. And we're going to follow them for some set period of time. Or we're potentially going to put a maximum on, while we're monitoring them. What is the length of our longitudinality? And so this is sort of our entry cohort. And then once we get our entry cohort, we'll go ahead and start measuring outcomes. And so when we do that, we're going to look for outcomes that occur during that exposed time. And these outcomes are going to be anything in our tree. So if we're looking at potentially 8,000 outcomes in our tree, we're registering all of those, potential 8,000 outcomes. And then typically we want to set an event incidence. So we want to make sure that when people have an event, that we're counting that event only once rather than several times if they have follow-up. So a lot of the data that we use for longitudinal database is claims-based information, administrative information, where someone can be followed up close in time. So say a week later, you go back to your PCP, your primary care physician, and you see somebody or your general practitioner. You don't want to have that second time that you've gone back for a follow-up count multiple times. So this is a way to ensure that when you are measuring these outcomes, you're only counting each episode of an outcome once instead of counting it multiple times over the course of the analysis. And so here, again, we have a toy study here just for use to show everybody how it works. We have a one-to-one match design. So we have, our exposure is going to be drug A, our comparator group is going to be drug B. We've assumed a very large population that might not be typical in order to just illustrate some components of the method here. So we've assumed 5 million exposed persons in each study group, which is pretty large. We're using a tree of ICD-9 clinical modification codes. So it's about 8,000 codes. And that tree file is what you downloaded if you downloaded the materials for today. And we put in 2 simulated signals. So we've taken this simulated data set and we forced in a signal at acute coronary syndrome at a relative risk of 1.5, so that I would consider that a modest relative risk. And then we've done that same level of a signal, 1.5, but we've done it for Stevens-Johnson syndrome. And the reason for choosing these 2 different outcomes is just to illustrate the differences between something that happens fairly commonly, so acute coronary syndrome, has a much sort of more frequent event rate than Stevens-Johnson syndrome. And so the idea here is to show that for the same relative risk, what's the difference in behavior when you have something that occurs very commonly versus something that occurs pretty rarely. And that's why we've done what we've done today. One thing to point out is that with 5 million persons, certainly, these are relative measures, and you'll see this when we go through the exercise from an absolute or public health perspective. If you have a relative risk of 1.5 in a very commonly occurring event, you have a big problem on your hands. So you have a lot of excess adverse events for a very common event, whereas a relative risk of 1.5 for something that's very rare, your public health burden is much lower. And so you should see that today when we go through, but just acknowledging it here at the outset. And so we're going to look at the orientation of the interface, starting with the analysis tab, which has information about your design decisions and advanced features. There's the input tab, which has information about the counts that are needed in the tree. And there's information in the output where you put your results. And so here I'm going to go through each of these things in turn. In this analysis tab at the start, we are going to use a fixed ratio match analysis, as I talked about, which is compatible with an unconditional Bernoulli probability model. And so what we would do here, and what I encourage you to do if you're following along at home, is open up your TreeScan tab or your program if you're doing this with me, and to go ahead and go over to your analysis tab here. So right now in the type of scan we're selecting, we're going to be looking at a tree only scan as opposed to the time elements. Time elements become more important in a self-controlled design when we're looking for the time to event that happens, or the timing relative to the exposure is really important. Here we're going to do an unconditional analysis with the Bernoulli model. We're going to use that Bernoulli model. We aren't doing a self-controlled design, so we're not going to click this checkbox. And we have to tell TreeScan what is the case probability in the underlying data set. So what that means is what's the fixed ratio. And so in a one-to-one analysis, you're sort of half likely to be in the treatment versus the comparator group. And that's why you get that case probability here of a half. So that's a really important point. When doing the fixed ratio matched analysis, is that the null hypothesis assumption here is that events are going to occur in relationship to the population of these two cohorts at the time. And so if they stay in a fixed ratio match, then there always should be an equal likelihood of an event belonging to either group. That becomes, of course, 1 out of 3, for example, if you have a 1 to 2 match. So I think you can see where this is going. But that's what you need to fill out here in the analysis tab. And so here, if you click to that advanced, so that's this little button in the bottom, if you click on that, and then within the software, you go into this other window that opens up, and it has several components here. One of the components here is about the inference itself. And this goes to that point of where that multiple hypothesis testing control comes in. And so the Monte Carlo replications are the way that threshold is set for where you're going to believe that any observation you make is going to be much different than what you expect. So sort of where your observed versus expected greatly exceeds, what you're looking for. So that one, you basically can fill out, the more replications you have, the longer your analysis will take to run. But you can also detect sort of more finer points of differentiation between different potential signals. Also, you're going to check here for the tree levels, and we're going to say we're not going to evaluate these higher level tree levels 1, 2, and 3. So one of the reasons that we do this is because some of the high level tree levels, if you think back to the tree diagram that I showed at the beginning, diseases of the nervous system, diseases of sense organs, typically that's not actionable. So doing a hypothesis test up at that level, while it won't spend that much of your alpha, it's a hypothesis test that if you can't act on it, you don't want to do it, essentially. And so we're choosing to do a hypothesis test at just the 4th and the 5th level, which allow for one set of clinical groupings and then the base term. The equivalent of this in MEDRA would be doing hypothesis testing at only the preferred term and the lower level term. And this is changeable, right? So if you decide that when you look at this, well, you think you really need to add in higher clinical groupings, you can then allow hypothesis testing at the 3rd level, for example, which would be the equivalent in MEDRA of saying, I'm going to allow higher level terms to be investigated, for those who are familiar going back and forth with the MEDRA terminology. So here in the input file section, if you tab this over, you basically are telling the software where your 2 files of interest are located. So here you want to put in on your computer, wherever your directory path is, you're keeping this file, and you're going to put in here the tree file itself. And so here in your count file, this is again your analytic data set. You're going to put in the directory path here. I renamed this to 2021 for our training today, but I didn't take a new screenshot of this particular screen, so I apologize for that. But essentially, you're going to point your software to where your analytic data set is, and that helps. That's just needed in order for the software to run. So really briefly to go through what these files contain. So in the tree file, the way that it is situated is it is a 2-column file. The left, and it's comma separated. So the left column here is what was known as the child node. And the right column here is known as the parent node. And so in a 5-generation tree, there are multiple children and parents, and you can think of it as just going up the generations. So if you're in a particular place in the tree where you are not only a parent, but also a child, it will appear twice. The base level code are the children for which they have no other children. So those are sometimes called the leaf level or the root level, or excuse me, the leaf level, or like the finest grained piece of information. So this would be the equivalent, again, for those of you who are more familiar with MEDRA terminology, this will be where your lower level term would go. And then the parent to that is there. And so this is just telling the software program what the structure is of the data so it understands how to think about the data. And then the analytic data set itself has 3 columns. And so in the first column is, again, that lower level node or the leaf level. And so this is a ICD-9 code, which has the decimals compressed out of it, which is what you see here. And then that second column is the number of outcomes that have been measured in the treatment group. And the third column is the number of outcomes in the referent group. And so essentially, what the software is expecting is that under the null hypothesis, because any outcome is equally likely to occur because of our fixed ratio match, that these numbers are relatively the same. So you see here for this particular node, 564, and this would be 0.4 if the decimal were not compressed. There is essentially approximately equal number of outcomes here. And you can see that there's some degree of stochasticity. It's not always exactly equal, of course, because of just the problem at hand. And so what it's going to do is look for places where there is a severe imbalance, and we'll walk through that here today. And so here is a visualization of the overall data set in our simulation. So in the overall data set, and if you look at these two treatment groups and you look at the total number of counts, because we've simulated there to be a null effect in essentially all the outcomes except for a few chosen ones, the overall data set looks as though there's approximately equal counts, which is what we would expect if we were under the null hypothesis. And then this is a visual representation of one of the two nodes where we've chosen to create a purposeful imbalance. So we've injected risk. And you can see here that we've got many more outcomes in our treatment group than our comparator group. And our hope is that the software then finds that imbalance that we have purposely injected so that we can see that difference. But the number of the imbalance in any individual node or individual outcome is not typically available to be seen in this overall data set because of I'm going to use a word that Nicholas used before, a masking effect, but it's in a different context. Essentially, it's just not rising to the top enough to be able to see it in the overall data set. And so finally, the last tab here is the output tab, and this indicates where you want to park your results. So this is on your own machine. You want to put in here wherever you want to find your results, whether it be on your desktop or in your My Documents, etc. And then here, I'm suggesting that you check these two boxes for additional reporting that is more convenient to look at. The HTML because it's visually more appealing, the CSV because it's used more easily when you're aggregating data for other purposes. And then also, this has an advanced tab in the same corner, just like the prior tab. And in that advanced tab, I'm going to suggest that you check these boxes to, one, report the attributable risk. I think it's important, again, especially when we're thinking about the differences between these two types of outcomes, one being very common, one being more rare, to look at the difference in attributable risk. You'll see it quite obviously, so that you're not thinking only relative. So sort of having an absolute and a relative, I think is helpful. And then reporting out the log likelihood ratios so that you can visualize what the test statistic looks like. And so alerting thresholds. So what is an alert? And so I know Greg used this terminology as well, calling something an alert. And then when an alert sort of stands up over time, calling it, then we sometimes graduate that to the signal. But what an alert really means is that we have a statistically significant finding of greater than the expected occurrence of the outcome among the treatment group as compared to a referent group. And it's user specified. And in general, any alert requires triage. So in Greg's presentation, you heard that discussed as, I believe, signal validation is the word he used. You've heard Nicholas, I think, in his presentation, use the word signal assessment. Here, I'm just going to say we're going to take an alert and we're going to see sort of if it holds after we do a lot of background information, in which case it might become a signal. And so I know that the terminology is a little confusing because it's different across jurisdictions, but it has the same aim, which is to see if something really is worth following and doing more work with. Some of the questions we could ask is this a labeled outcome? Is it something that we expect? Are there case reports in the literature? Is it a non-specific outcome? Does it represent viral signs or symptoms that we really can't do anything with? And so those are some of the things that we can look at when we try to decide whether we're going to continue to sort of delve deeper into this particular component. And so here, right now, if you were to have essentially walked along with me, the analysis tab, the input tab, and the output tab, at this point, you can run. And so this would be the part where, if you want to allow TreeScan to run in the background, it should run very quickly. It's not a very large data set. It should finish up in something like 3 minutes. So if you want to run in the background now and get this to pop up on your own machine, please go ahead and do that. But I will go through the results as though we have already run it at this part. And so here, this is just to look at some of the language here. The total cases are an indication of the number of outcomes that are observed in your treatment group, whereas the total observations are the outcomes that are observed in both your treatment and your comparator group. And I've just done the quick math up here to show you that we're running about 50-50, which is what we expect under our simulated problem, because with the exception of a couple of outcomes where we put the risk in, everything else has been set to null. And so this is as expected, the ratio of, again, the cases in the treatment group to the total number of cases in both the treatment and the control group. And you can see these are fairly large numbers, as you would expect, because our underlying population we simulated was 5 million people in each exposed group for 10 million total, exposed to either the treatment or the comparator. So quite a large population that we've simulated for the purposes of showing this today. So some of the other information here, this is sort of just details about the tree, the total number of nodes in the tree, the root nodes, the nodes with children, the leaf level nodes, and then the number of levels in the tree. And then this is showing you as you go up. So again, to do that sort of look back to measure terminology, this level here, the leaf level, is the equivalent of the lower level term. This is the equivalent of the preferred term. This is the equivalent of the high level term, and so on and so forth as you go up the tree. Each time you're getting to these larger clinical groupings of ideas. And so this is what the output of TreeScan looks like. And so this is the output of post-analysis, our data set. We actually saw the 2 alerts we expected to see, meaning that for our simulated risk, TreeScan is able to identify them. And so here, if you recall, the acute coronary syndrome is node 411.1. So again, the decimal is compressed there. It alerted at the tree level, meaning that in this case, the leaf level, the individual outcome is the 5th level. And then for in this particular tree, there is no difference between the next higher level grouping. And this is a code for the next higher level grouping. And you can see that that's the case because it has identical numbers. You'll also see in the HTML file I show you later what the mapping looks like of the tree. So in either way, you'll be able to see that particular component. And then here, the third sort of it's listed in the software as a cut, but what it means is sort of the things that are the most unusual in your particular analytic data set. So here, this is the compressed code for Stevens-Johnson syndrome, 695.13, and it's showing you again that it's an outcome level node. And so first off, you can just take a look at the number of outcomes that you see. And again, this is a combination of both your treatment group and your comparator group. And we used as the seed for this entire problem, real outcome rates. And so that's why you see these huge differences in terms of the total number of outcomes, because the difference here between a more frequent outcome of acute coronary syndrome and the Stevens-Johnson syndrome is just the volume of caseload is quite a bit different. And so next, we're going to look at the number of outcomes that are particularly in our treatment group. So again, when the log likelihood ratio, when TreeScan looks at this, it says, okay, so of all of these observations, I expect all these observations that they're equally likely to occur in my treatment group or my comparator group. And then it looks and it says, okay, these are the observed cases in my treatment group, but my expected, you'll notice, is exactly half of this total, because it's expecting that if I had 38,480 outcomes overall in this node, that half of them belong to my treatment group and half belong to my comparator group. And then the comparison of the observed to the expected is what you see here with the relative risk. And here I'm showing you a snapshot of the user guide, which shows you the mathematical formulas used to calculate the expected value, the relative risk, and the excess cases for every type of scan available within the software. And so if you ever have any question about what this number means, you can refer to the user guide and to the mathematical formulas to find that out. And here, similarly, as I was showing before, the excess cases are shown here. So the excess cases here are just the subtraction of the observed minus the expected. And then you put an attributable risk on it where you're looking at it per how many people exposed in your treatment group. And this is what I was talking about before, where the particularly common outcomes, you're looking at 2 excess cases per 1,000 exposed persons. But in the fairly rare outcome, even at the same relative risk, you have a very large difference in your absolute or your attributable risk, where you have 8.3 excess cases per million exposed persons. So just illustrating that even for the same relative risk, you have this large difference in attributable risk based on the underlying event rate that occurs. And then here, this LLR is the log likelihood ratio. Again, we're in a likelihood ratio test method. We use the log likelihood ratio for mathematical convenience, but essentially a log likelihood ratio and a likelihood ratio, you can think of them as they're more or less interchangeable. They have properties that make it easier to use one or the other in various circumstances. When this number is high, what it means is that, and I'll show you this in a couple of slides in the math, is that the departure from the expectation is very large. And that departure from the expectation is built around 2 different components. One is the actual relative risk, but the other is built around the overall size of the data set. So how much contribution do you have? And that's why you can see that for the same essentially relative risk between an injected risk for a common event versus a rare event, Stevens-Johnson's, there's a huge difference in the departure from the expectations. And the one that's larger is the one with the greater power, essentially the greater sample size. And so there's a sample size component to this in addition to the actual, the relative risk ratio. And so here, the p-value here indicates where in the distribution the test statistic is going to fall. And so I'm going to show this in a slide visually in a minute, but I'd like you just to remember for numbers wise that our log likelihood ratio for our very common event of acute coronary syndrome is about 5,200, whereas our log likelihood ratio for our less common event is about 14. It's going to be important just on this next slide in a little bit. So I'm actually even going to make a little note to myself to write it down here and come back to it. So this is how the test statistic is calculated. I love the math. I think the math is really helpful. I apologize for people who hate math, but I think it's insightful to think about how these numbers work and how we see this. And so this is a maximum likelihood estimation. It looks like a Bernoulli probability model for those of you who are familiar with probability models. The numerator here is based on the observed data. So these quantities here are based on the observations of the total number of outcomes in your treatment group and in your comparator group, whereas the denominator is based on your expectation. And that expectation was based on your fixed ratio match. So here the P is that probability of being in one group or the other. And so the bottom line here is that when you compare your observed to your expected, if you have essentially the null, then you have no departure from your observed or your expected, and this whole quantity goes to 1. And when that whole quantity goes to 1, the log of that is 0. And so essentially, you're not going to see any value for a signal at all. When there's a big difference between your observed and your expected, this quantity gets larger and larger. And the larger the quantity is, the larger departure from your expectation that exists. And so a couple of other components of this. This here is an indication function. What it means is that we're only looking for times when the observed exceeds the expected. And you can see that very similarly to when Nicholas had his great graphics where he was showing the stacks of the reports. When there is a stack of the expected that's greater than the observed, it's going to show up on no one's radar, because this is really only looking for when the observed is greater than the expected in terms of the stacks. And so that's the only time when you're going to calculate that log likelihood ratio. And then finally, the idea here is that this value is calculated for every individual position, every node on the tree. And that when, again, you have a large departure, you're going to show up with a very high node specific log likelihood ratio. But when we compare that, we're going to compare it to the maximum across the entire tree. And the reason for that is this comes into the Monte Carlo simulation component. We're looking for a threshold. And what we're looking to see is the difference between the observed and the expected so large in certain circumstances that it beats everything else out on the tree. The entire tree, this is a departure that is more significant, or not significant is an inappropriate word to use, is more important than any other node that you see. And so here is a graphical depiction of that. So this is when you do Monte Carlo hypothesis testing, essentially what you're doing is you're creating a bunch of data sets under the assumption of the null. And then you take the maximum again, and you use the same procedure, and you take the maximum log likelihood ratio and you graph it. And so, and you don't have to graph it. I like to graph it because I can see what's happening here. And so when I graph it, I'm looking at this distribution. And then I'm going to compare that log likelihood ratio for each outcome to the position on this distribution. And so if you recall, the two numbers that I wrote down that I remembered is 5,200 for the acute coronary syndrome and 14 for the Stevens-Johnson syndrome. So you see 14 actually even appears on our graph. So it is in, of course, the upper tails of this distribution, but it's actually still even makes it on the graph. 5,200 is like way, way off the page. It's so far beyond our expectation that it's basically screaming for someone to pay attention to it. And that's the whole idea of signal identification or signal detection, is to focus one's attention on where are the places where I have to look for where there may be this imbalance in the observed totals versus what my expectation is. And so with that, I think we'll return back just to look at these again with the 5,200 and the 14. So if you can think about placing those on that distribution, whereas the greater we are in the tails, the more worried we are that there might be something actually going on that is worthy of our time to investigate. Here is a visualization of the tree. So this is what I was talking about before, that acute coronary syndrome is a leaf level code, but it's the only one, for whatever reason, in the structure of the tree that feeds into this clinical grouping. That's not always going to be the case, but it was the case in this particular instance. And so now this is the part where, if you've been following along, we have an exercise to do, which may take some time, and which I might circle over to the chat and see if I open up the chat and we go ahead and do this. And the idea here is, if you wanted to perform signal identification yourself, if you've been following along and we've gone through the demonstration and now we're in the exercise, the idea here is to open up that analytic data set, which is that file that ends in CAS. And I would recommend, if you do this, that you open it in a text editing program. I wouldn't recommend Excel because Excel tends to drop leading zeros and has a bunch of other things, or it reformats data in a way that is undesirable. So if you can open it in a basic text editing program, that's a better way to go about doing it. And the idea here is to see if you change the analytic data set, if you can force a signal or not based on what changes you make. And so here, I think the important thing to think about, again, is about relative sizes. So say you wanted to add in some extra outcomes into the treatment group in order to sort of force a signal. If you add 5 additional outcomes into something that only occurs sort of 10 times or less, you're basically creating a very large effect size. So you're putting a lot of excess cases into your analytic data set. And it should be very easy for TreeScan to detect such a thing. But if you put in a small number of additional outcomes into something that occurs 50 times, that's going to be much harder to detect, because it's going to be much harder to create that difference. And so the idea here with this exercise is just to give you an intuition or a feel for how the mathematics and how this works, so that you can detect something using your own data set. And so what I'd encourage you to do is, if you do change your analytic data set, that you save your new file with a new name, and then go through and then run TreeScan again. And so I'm going to pause here and open up the chat, because I haven't been looking at it, and see if anyone has any questions. Oh, I see none. So that's maybe not that great. Or see if anybody would like to talk about how one might go through an exercise like this. And this is the part, Holly, where I was going to say, if we can let everybody unmute themselves, we have a small enough group that if people are participating in this exercise, it might be helpful to allow them to speak at this time so that we can answer questions as they go. But I'm going to give a couple minutes, and maybe it would be helpful if people, if there's a way, we don't have a poll here, but if you want to write in the chat, if anybody's doing the exercise and want some time to go through it, or talk out loud. So Holly allowed us to have everybody talk, so we can talk about how one might use this type of an analysis in your own work, or if there's any other questions that folks might have. Happy to answer. Also, if we're waiting for folks, if anybody is doing something, happy to have my co-panelists, Greg or Nicholas or Monica, if anybody wanted to add anything, or any callbacks. I think a lot of these techniques, as Nicholas I think pointed out, are all very similar. They're all sort of geared around the same thing. They're just slight variations on the theme. And so hopefully this gives you some intuition about how they work, similar to the intuition I think was being developed earlier with the visuals of the number of reports. So I see a question in the chat. Can you talk more about the distribution selection? How do you know which to pick? I think maybe if we're talking about Poisson or Bernoulli data, sort of which likelihood method to use. That really has to do with how you've collected your data set. So I'm going to just go back in and hoping that this is the right question. But if it's not, please feel free to type in the chat if I'm not getting at the right question. Here, I mean, the main thing to sort of take away from this is that in a Poisson type study, what you're doing there is you're taking your treatment group outcomes, and those are happening, in real time. Your comparator group or your referent group in a Poisson-based analysis can be historical, it can be concurrent, but essentially what the difference is that it is a rate, it is one value, and you use that value to then set up your observed versus your expected. And so, the parallel of this to the spontaneous reports is the value of this in spontaneous reports is the overall, sort of rates of the particular reports in the comparison. And so that could be the rates of, all drugs and all outcomes, or it can be the rates of all drugs and all outcomes, subgrouped by adult reports or something like that. And so here, the idea, I think the main thing to think about is that you are doing more of what I think of as a one group monitoring. So one group is active, and the comparator group or the referent group is just a value. It's not really a stochastic quantity. The difference here in a Bernoulli sort of setup is that you have two different groups you're monitoring, and both of them have some stochasticity associated with them. Both of them are having reports come in, or in this case, outcomes at the same time. Which part of the package will be able to account for the propensity score? So the propensity score has to be done earlier than the analysis. So at the time when the analysis is done, the analysis is just to go back to the analytic data set to illustrate this. The analysis here is really the outcome, and then the total number of outcome, or the outcome itself, the total number of outcome counts in the treatment group, and the total number of outcome counts in the comparator group. And so all of the design controls that would be available in the propensity score design or an exact design have come in to get to this analytic data set. So essentially, the 119 and the 105 are based on, in this case, having done a propensity score matched analysis previously and only allowing the outcomes of our matched population to be brought forward. So outcomes, for example, for the unmatched population would drop out. They would never make it into this data set. And so that really is the component of where the design piece comes in that your design already from the start has set up what actually is in your analytic data set. And then this is this part, the software that you see here is just doing that last component. So the propensity score piece or the exact match piece, all of your design-based confounding control needs to be done at an earlier point before you get to here. And if this Bernoulli set is compatible with a propensity score match design, but a different, the one I showed earlier, the Poisson, that's actually compatible with a propensity score stratified design. The difference here being that in a match design, there has to be an underlying understanding of what the value is of the relationship between the treatment group population and the overall population. And in the Bernoulli design, that has to stay fixed over the course of the analysis. Whereas in a stratification type analysis, it doesn't have to stay fixed, it can vary. And that's one of the differences in design and how that then feeds into the method. Keep in mind too, maybe an important point here is that, again, say you get the alerts that we've gotten here that come out of this. What does this actually mean? So in this case, for acute coronary syndrome and for Stevens-Johnson syndrome in this case, we forced a situation with simulation where we've caused these signals. But really, what am I taking away from this? Going to that triage step, I'm saying that there is an unusual elevation of cases of acute coronary syndrome or Stevens-Johnson syndrome in my treatment group relative to my comparator group. And in the case of a propensity score match design, it would be drug A compared to drug B. And so then the question is, why does that happen? And in the alert triage, it could be that, again, that could be a labeled event. That's such a large difference. The idea if it was labeled is that maybe people wouldn't actually be experiencing it in those numbers, but you'd be asking those kinds of questions. And important in the propensity score itself is that, back again to something I said earlier, when you're in a signal identification analysis, the propensity score has not been geared around these particular outcomes. You could end up with a different result in an evaluation study if you changed your propensity score to then be focused on these two particular outcomes. Because if these were your hypotheses that you generated from your signal detection, so now let's talk about just one of them. We'll talk about Stevens-Johnson's. So, you generated a hypothesis that drug A is more associated with Stevens-Johnson syndrome as compared to drug B. Then if you were going to design a study, you might want to add a lot more into your propensity score that deals with specific risk factors around Stevens-Johnson syndrome. And that newer propensity score is different than what was used here because it's much more targeted. And so there you've set up a different design. And so some of the things a lot of people think about, which I didn't prepare necessarily slides to talk about here, but since I'm not seeing any technical questions come in, I'll answer it and then maybe take a pause and we can go to Monica's talk. I have to do a time check. But one of the things a lot of folks are thinking about is, well, what about the data that you've already used for this signal identification analysis? And oh, by the way, you already used a propensity score. How do I then set up to do a epi study? Can I not use that data set anymore? Am I not allowed? That kind of thing. So there's been a lot of work that's been done thinking about that, well, do I go to a new data set if it's possible and try that? But another thing that you could do is do a different design. Do an orthogonal design is what it's called, where you're not actually, you've adjusted your study question. In this case, you can adjust your propensity score. So you're actually doing a different analysis. and then look at it that way in order to build more information around what you see from this signal identification analysis. So I'm going to take a look quickly at my time, and I think I am up for time. I think we are ready to transition to Monica's talk. Is there just last call for questions? And then Holly, if you would mute everybody again, I think they all look muted anyway, just to make sure that Monica has the same situation that we all had. That would be great. Thanks for your time today, everyone. I hope that if you participated in the exercise and if you have any questions, you can go ahead and send me an email if you have any thoughts. Thanks so much. Monica, over to you.
All right. Are you seeing my screen? Hopefully.
We are. Not in presentation mode, but now, yes.
All right, cool. So thanks, Judy. I'm Commander Monica Munoz. I'm currently the Deputy Director of the Division of Pharmacovigilance 1 within FDA's Center for Drug Evaluation and Research. I'm a pharmacist and pharmacoepidemiologist by background. I started in pharmacovigilance at the FDA as a safety evaluator conducting surveillance of neurology products. Then I led a team in the area of cardiology and nephrology products prior to becoming the deputy director. In the deputy role, I provide scientific and some administrative oversight for many of the division's pharmacovigilance activities and initiatives. You may also notice that I'm wearing a really cool uniform, and that's my other literal hat as an officer in the U.S. Public Health Service. In that capacity, I've had the opportunity to respond to many public health missions and deployments and to respond to things like hurricanes, Ebola, and of course, most recently, COVID-19. So that's a little bit about me and my perspective. At this point, Greg laid down the foundation for pharmacovigilance, signal detection, and its importance. And Nicholas and Judy have described different quantitative techniques that can be applied to identify signals. The goal of this talk is to pull things together and give a practical perspective on what happens after we detect a signal. So I'll provide a practical overview of how case reports are assessed beyond signal detection, since these reports are really the cornerstone of pharmacovigilance. And then I'll go through an example of a real case study to illustrate how information is synthesized to inform actions aimed at improving safe use of medicines. So FDA Center for Drug Evaluation and Research is responsible for conducting surveillance of all approved and unapproved drugs and therapeutic biologic products, monograph or over-the-counter products, compounded products, homeopathic products, so really thousands of products. The pharmacovigilance activities begin pre-approval and continue throughout the life cycle of the drug, which we'll illustrate later. And the approach to pharmacovigilance is very multidisciplinary. The different disciplines involved really depends on the safety issue that we're looking at, but typically involves different clinicians, statisticians, epidemiologists, sometimes even engineers, chemists, depending on the issue. And then we use a risk-based approach because we surveil thousands of products. So we generally focus on products where less is known about their safety. We'll prioritize the evaluation of risks that can significantly impact the risk-benefit balance. And our priorities can change over time as drug information accumulates. So I want to cover signal management at a high level. And when I refer to signal management, I'm really talking about all the activities that revolve around a potential safety concern, from the identification or detection to refinement, which others might call validation, to the evaluation or the assessment, and for FDA, taking a regulatory action if one is warranted. This framework starts with signal identification activities we've been discussing for FDA. This is our ongoing surveillance of different data streams like FDA's adverse event reporting system. That's our spontaneous reporting database. literature, periodic safety reports submitted by sponsors, and safety concerns really from any data source. We may get them from other regulators, other submissions to the agencies, and so forth. The signals from any source move to signal refinement. Here we're still in pre-evaluation, and we're considering the preliminary findings on the causal relationship between the product and the safety concern and any potential health impact in the available data streams that we have, which usually includes considering data sources beyond where the signal originated from. This preliminary assessment of the evidence informs the triage and prioritization of signals that I'll talk a little bit more about in a minute. Then we move to the evaluation phase. This is the detailed assessment of the evidence by a multidisciplinary team. And based on the evidence from the signal evaluation phase, the agency will take an action, which may include further assessment, communication, and other risk minimization activities or actions in accordance with the importance of the issue. So the pre-evaluation phase is all about figuring out, is there a signal worth investigating further? And there are lots of activities that can inform this issue. The examples listed here are from FDA's manual of policies and procedures for newly identified safety signals that you can refer to. But say we observe a disproportionality in spontaneous reports or in the electronic health data for a particular drug event. We then want to look under the hood and look at some of the characteristics of the underlying patients. We may look at a sample of reports to see what they're really describing and so forth. And all of these activities will then inform the triage and prioritization of the signals. So as we've said earlier, and I think Greg highlighted all the different possible sources nicely in his talk, that a signal can come from any source, and we review them in different teams that are specific to certain therapeutic areas and have that body of knowledge to judge these signals. So the team ultimately has to decide, are we going to evaluate this, or can we refute it, or are we going to continue to monitor the signal? And there's lots of different factors that we may consider. So, first of all, is this really a new adverse event, or, is it something that's labeled? And even if it's labeled, is there some new aspect about it or some increased severity about the particular adverse event? What's the severity of the adverse event? What do we know about the products? How is the product currently being utilized? Are there particular populations that are vulnerable, exposed, like pediatrics? And how many people are exposed to the product generally? Then we have to consider the strength of the evidence. So do we have an idea of the magnitude of the risk, and what data source did that come from? Is it from a really well-designed observational study, or is it from a few case reports describing something that may be difficult to distinguish from the background of the disease and the treated population? And is the public aware of the potential signal? There may be a need to formally evaluate the signal and communicate the findings to the public, even if the evidence supporting the signal is weak, particularly if there are implications on adherence or prescribing potentially beneficial medications. We also have to consider the reality that all pharmacovigilance organizations have finite resources that necessitate that we prioritize signals. Evaluating a signal means you're going to bring together the experts to determine if the signal actually represents a risk, which means those same experts aren't working on a different signal or performing other safety activities. So if we have 10 signals, which are the ones that have the greatest potential to negatively impact public health, we want to work on those sooner. And it can be pretty challenging to prioritize signals, but the factors bubbled out here are ones that teams routinely consider to inform this decision. Now I want to switch gears a bit and talk about case reports, because as Greg said earlier, these are still the bread and butter of pharmacovigilance and will probably continue to be. and a retrospective review of the FDA's internal files of all labeling changes that were safety related that occurred in 2010. This was about 400 drugs. Spontaneous reports contributed to over half of those labeling changes. We recently completed another analysis of more recent data and know that this is still very much the case. Whether they're case reports from fairs or a sponsors database or the literature, these reports are still driving the majority of labeling changes a decade later. The contribution of the different data sources is certainly dependent on the adverse event. Events like serious cutaneous adverse reactions or SCAR are very much in the wheelhouse of case report data. Judy talked about Stevens-Johnson syndrome. So Stevens-Johnson syndrome and toxic epidermal necrolysis are the most concerning SCAR because of their significant morbidity and mortality. They're often drug-related. The onset is usually a week to 4 weeks after exposure. And in the general population, there's a few cases per million people. So a rare event, severe, relatively short time to onset, right in the wheelhouse of case report data. So we recently looked at all labels that had a SCAR added to the labeling within the past 5 years and what their underlying data sources were, as well as some other things. We looked at the language added to the labeling, but all but one were triggered by reports. I think one was triggered by an observational study. So that's obviously the more extreme contribution of case report data to labeling. So now we're going to dig into how we review case report data after we've identified a signal. Then we'll move into integrating case series findings into with the context of other information and ultimately determine what actions, if any, are needed to mitigate any identified risks. To develop a case series, it's really a stepwise approach, and I'm going to present this as a totally linear process, but sometimes you may complete multiple steps simultaneously depending on what the data looks like. We usually start with a case definition, then we'll identify cases of interest from usually fairs and the literature. We'll screen out reports that we aren't interested in, apply the case definition, assess causality, and then we'll have this set of cases that we'll call our case series, which is usually much smaller than the initial batch of cases that we started with. The case definition is usually developed with information from the medical literature and clinical practice guidelines and consists of criteria like signs, symptoms, diagnostic tests that define a particular disease, injury, or health condition. And using case definitions provides a consistent and reproducible approach to identifying particular cases of interest. A good example is Sampson's criteria for anaphylaxis that's often used for assessing case reports or adapted to assess case reports. The criteria specifies that certain systems must start within minutes to hours that include skin and mucosal involvement, plus respiratory, hypertension, or other signs of end organ dysfunction. Once we have a case definition in hand, we then need to identify the cases that we're actually going to apply this to. And this slide captures a few pearls of searching fairs. And it's not intended to be comprehensive, but adverse events in fairs are all coded using MEDRA terminology that was discussed previously. If we think about our anaphylaxis example, we can look for PTs that correspond to the different aspects of our case definition, like rash, hypotension, and these are all actually captured under one standardized MEDRA query. You also need to search for whatever product or products that you're interested in. In fairs, all products are mapped to product active ingredients. So if you search for a generic name, you will also pull any reports that were reported under the brand or the trade name for the product. In the context of signal detection and evaluation, best practice is to not limit your search to just primary suspects. Will say that in my experience, the coding is often arbitrary and may, the primary suspect may not actually be the suspect the product or the reporter thought was the most likely to be implicated for the adverse event. And then one last pro around generic and quality issues that we frequently get questions about is when a product has generics on the market, it can often be difficult to attribute an adverse event to a specific manufacturer. Sometimes drug utilization data may help put this in context or give you some clues that it may not exactly be what you expect. So be careful in assuming that ascending manufacturer is the actual product manufacturer in the database. So we've searched the database and have a batch of reports in hand based on our search criteria. And now we want to review these reports. One thing that we often do is look for duplicates. And what I'm referring to here are cases that describe the same occurrence of an adverse event in a particular patient that may be sent to the database by one or more reporter. They're not uncommon in FAIRS. We often observe them when there are multiple suspect products, when people are on multiple combination therapies, when generic products are available. So each manufacturer may identify a case and send it to the agency. And we particularly see this a lot with literature cases. As Greg noted, a lot of companies are doing surveillance of the literature, and when they become aware of cases in the literature, they have reporting obligations to send them to the agency. So I would also note that duplicates are often a reason why you may see a disproportionality in reporting. So they're usually not too hard to spot once you look under the hood at the actual data. I'll also mention that there are some tools that help identify duplicates, but it's still a pretty challenging area to automate if you dream of cleaning them all up out of the database. We're looking forward to you doing that. But there are some additional papers cited here if you're interested in duplicates and the challenges around that in spontaneous reporting systems. So I'll show you a couple examples, and this is data from the FAIRS public dashboard. Here I've searched for clobazam, toxic epidermal necrolysis, and an outcome of death. And there are 15 cases if you go to the dashboard and repeat the same search. And I've sorted this conveniently to group some cases that I know are duplicates. So if we look at some of the characteristics of the cases, the first case seems to be not have much information in terms of demographics, and it's probably a unique case. But the next 7 or so cases describe a 77-year-old female who have an event in April or March, and all from Germany, and all have different sending manufacturers. And you'll notice here, the first product here is the primary suspect, but all have different primary suspects here. But these are really describing the same case. And we see another batch with a 66-year-old male, and then one last batch. So really, we retrieve 15 cases from our search, but we're talking probably about 4 unique cases. Here's one more example. I mentioned that literature cases are often a source of duplicates. This is one particular case of a 2-month-old infant who experienced toxic epidermal necrolysis. And you can see it comes out in the database over 10 times from a bunch of different senders. So sometimes easy to spot, sometimes difficult. The more unique the report is, particularly for that treated population, the easier they are to spot. If you're looking at embolisms in patients on contraceptives, everybody's a female of a certain age and have similar presentations, then it can be actually more challenging to identify true duplicates. All right. So we've removed some duplicates, and one thing that you may want to do is screen for quality reports, right? Greg harped on quality, and I'll do the same, that, you know, we really need enough information to assess a case definition and to assess the relationship between the drug and adverse event. And given the voluntary nature of reporting and few mandatory elements, the quality and quantity information provided is widely variable. There are some tools that you can use to measure this. The best known is Vigigrade, which has several adaptations in the literature that looks at the completeness of different data fields. It's pretty easy to automate scoring and adapt to different surveillance databases. In practice, the best tool is really just reading the case from a practical perspective during case review. And the tools like Vigigrade are great for providing a quantifiable metric, and they have been used for applications relevant to PV systems, like has the quality of reports changed over time, or are the differences in reporter types. But in practice, quality determination is frequently done by a cursory review of the data provided within each case. So here I have an example of two cases. Case one has missing demographic information, no real timing information. The reaction description is limited to a rash, medical history, other exposures, all these things unknown. And, you know, I would say this case is in the unaccessible camp. And case two is really the opposite. You know, we have much more specifics. We have specifics on timing and medical history, and, you know, some causality can be assessed, as well as the case definition in case two. At a higher level, we know quality is variable, and there are certain pockets in the database of reports that have lower quality of information. And I'll just describe one example here. And this comes from the fact that the FDA receives a lot of inquiries about cases with an outcome of death in the database from researchers, from the media, patients, where they look up a particular product in the database and notice a high number of cases with an outcome of death in comparison to what they may expect to see. But the reporting patterns observed are often driven by reasons other than new drug safety concerns, such as the underlying population's comorbidities, litigation, poison control centers send us reports, and, you know, we've seen some examples of extreme duplicates. So in our experience, we also noticed a growing number of reports that were coded not only with the outcome of death, but also the corresponding preferred term or an adverse event of death, and only that adverse event. And when looking at these reports, we felt they often contained limited information. They basically said, you know, a patient on an unknown date and unknown time after possibly being on this drug died. And that's really all the information you had in these reports. So we characterized and took a random sample of reports that had death as an outcome with only death as a PT, and compared them to another random sample where some other adverse event was reported. It could have been pneumonia, it could have been pain, really anything, and found that the death-only PT report contained much less detail than the coded fields, as well as in the narratives as well, much less information. So anyway, this is just an example of there are these pockets of reports that may differ throughout the database, and you may not know until you dig into the data a little bit. All right, so now we're at a point where we've removed duplicates, poor quality reports, and applied our case definition. And we need to assess causality. And causality assessment is performed to evaluate the likelihood that the adverse event is caused by a specific drug. Although originally proposed for epidemiologic studies, Bradford Hill is still a great foundation for assessing causality and pharmacovigilance. And a couple of references here are adaptations of the Bradford Hill criteria and mapping it to PV kind of terminology. Here I've grouped concepts into intrinsic and extrinsic criteria, with intrinsic meaning data gathered from information within that particular case or individual patient level data, and extrinsic can often be thought of as data from other sources, whether that's literature or the reviewer knowledge. And we talked about some of these aspects previously: chronologic data, what are alternative etiologies, any precedents or AEs with the same product or related products, biologic and pharmacologic plausibility are all things to consider in the assessment of the case's causality. Generally, this requires reading the narrative and application of clinical judgment. There are different methods that have been used to apply the causality criteria from the last slide, just global introspection, algorithms, probabilistic or Bayesian approaches, but there hasn't been a method that's demonstrated to be superior. There are also tools that may be used for specific adverse event causality assessments, like the Drug-Induced Liver Injury Network has a tool to assess liver injury cases, but no tool is really proven best. And often you may take a team approach to looking at certain challenging cases to get some perspective on what you think the actual causality is. So I mentioned that there also have been attempts to try and automate different parts of case series development, and the papers here are aimed at identifying cases of interest, particularly cases that were either included in a case series or were determined to have at least a possible causal relationship. And ultimately, I think what we learn from these projects is there are opportunities for advanced tools that use machine learning or NLP and other techniques to assist with case assessments, but performance of these tools to date is more in the supportive or decision support realm rather than automating aspects of case assessments, particularly causality assessments, which are particularly challenging to automate. So here's the overview. We started with this stack of cases. We removed duplicates. We assessed the case definition and causality and ended up with a case series. And the numbers here on the slide represent the median number of cases at each stage based on about 70 reviews completed in 2016. The median yield is around 20%, but varies widely based on the drug and adverse event being evaluated. But it's clear that the number of cases you start with will almost certainly change as you go through the different steps of developing a case series. So you'll want to summarize after you have the case series what you're looking at, because you're now preparing to put it in the context of everything else that you know about this particular drug adverse event pair. This is an example of direct oral anticoagulants and cutaneous small cell vasculitis, and an example of what summarization looks like. So what do we know about the drug, the time to onset, what was the flavor of vasculitis, and so on. And sometimes these types of characterizations can inform follow-on analyses, like it did in this case, which observational study was done after the cases were reviewed. All right, so we have our case series, and we need to view it now in the context of all other data sources we have. And to illustrate this, I'm going to walk through a recent example that pulled together many different data sources and led to some complex decision-making, which is montelukast and neuropsychiatric events. So montelukast is a leukotriene type 1 receptor antagonist that was first approved in 1998 for asthma in patients 15 years and up. Over the years, the indication has expanded to include allergic rhinitis, and then in 2005, prevention of exercise-induced bronchoconstriction. actually in 2007. Over the years, FDA has conducted reviews of neuropsychiatric events since becoming aware of post-marketing reports in 2007, and these activities have resulted in successive labeling changes and public communications. However, there's been continued concern among stakeholders about the risk of neuropsychiatric events and a lack of awareness among prescribers and patients. So based on these concerns, the FDA initiated another comprehensive review of the issue, including conducting an observational study in fentanyl and holding a public advisory committee meeting to discuss the review and solicit recommendations from a panel of experts. This timeline here shows some of the regulatory history around montelukast and neuropsychiatric events. In blue, you can see the various labeling changes over time prior to stakeholder concerns in 2017, and then in white, you see some other activities, communications and advisory committees where neuropsychiatric issues were discussed. So for this review that was initiated after 2017, we wanted to take a comprehensive look at all the available data, again, to decide if there is additional actions warranted around neuropsychiatric events. And we had to consider the strength of evidence and the totality of data from all the different data sources. So we're going to walk through each one of these, and this is something you may do for most signals. And in this case, we really did formally evaluate each area and describe them in a review. So we'll start with clinical trial data. And the clinical trial data is actually pretty sizable given a number of different indications had been evaluated. However, there wasn't really a signal for neuropsychiatric events or suicidality with montelukast, but there were limitations to this review. The clinical trials were really short in duration when you consider what the indications are. Many of the trials are just a few weeks long, and they were not designed to examine neuropsych events. We also looked back at non-clinical data. Orally administered montelukast is detectable in brain tissue and CSF in rats, providing some evidence of its ability to cross the blood-brain barrier. However, the specific, receptor that montelukast is targeting is pretty low or non-existent in human brains, although it's certainly possible that there are other mechanisms or pathways to neuropsychiatric events. But overall, there was a lack of a clear mechanism for neuropsych events from both the clinical trial and the non-clinical data. FDA looked closely at the utilization data, and montelukast is a really widely prescribed product. In 2018, approximately 9 million patients of any age received a prescription for montelukast in the U.S. from outpatient retail pharmacies, which is the primary distribution pathway. Of these, over 2 million were children younger than 17 years of age. Most were prescribed montelukast for asthma, followed by allergic rhinitis, and then by cough, which was concerning given cough is a use that benefit hasn't been established. Numerous reviews of post-marketing reports have been completed since 2007. Many have informed the addition of various terms that I showed you in the regulatory history timeline as different reviews were completed. Overall, there is a wide range of symptoms and the severity of the events. Time to onset varied pretty widely, and not really clear risk factors to be identified from these case reports. And unfortunately, the background rate of neuropsych events like depression and anxiety and suicidal ideation are not uncommon and have increased in the U.S. significantly over the past decade, particularly in children. So with the background rates, the variability in presentations, the variability in timing, it really made it challenging to determine causality in individual cases, even though there were some compelling anecdotes that the agency received in case reports. The FDA also reviewed observational data. In the most recent review, there were 4 key studies prior to the Sentinel analysis that was initiated. They had mixed findings. The study that suggested montelukast, or most strongly suggested montelukast is associated with neuropsych events, had significant limitations and was really highly susceptible to recall bias. A couple of the other studies didn't find an association, but also had other limitations and may have been poorly powered. So FDA attempted to address some of those limitations with a new study in Sentinel. The study investigated whether there's an increased risk of hospitalized and treated outpatient depressive disorders, self-harm, and suicides associated with montelukast used for asthma as compared to inhaled corticosteroids. Overall, the risk of the events compared to steroids was not significant in the study. But again, there were some limitations to this analysis. Primarily, we were limited to events that actually recorded as claims. Most of the data was post-2008, where prescribing information and changes in communications had kind of been out there. And the investigators weren't able to adjust for socioeconomic status. And you can see the details of the study referenced in the paper here. Finally, the other important piece of evidence that the FDA considered was information from the Pediatric Advisory Committee. FDA uses these committees to obtain independent expert advice on scientific, technical, and policy matters, as well as to gain insights from various stakeholders like patients and caregivers. The comments we received from public speakers at the advisory committee, as well as through a public docket, was a strong sentiment to issue a box warning. Again, this emphasis that there's a lack of awareness of the potential risk despite labeling, and probably an unfavorable risk benefit for allergic rhinitis. Panel members had mixed opinions on a box warning, and this is primarily driven by the uncertainty in the data and this desire to have better data to evaluate the risk. But there did seem to be a disconnect between the data and the public comments, and they felt that the risk benefit needed to be reevaluated for each indication, and there continued to be a lack of awareness, even amongst the panel members, of the risks that were associated with montelukast. So now we're at a place where we've pulled the information together, and we're at the challenging part of what do we actually do with the information that we learned. And there are several actions generally that the agency can use to mitigate risks. One of the most common things we do is modify the product labeling. This could be adding a new warning. This could be changing the language in the labeling. And there are guidances and regulations that dictate, what goes where in labeling and how things are described to communicate risks. We could require a post-marketing study or a trial. We could implement a REM or a risk evaluation mitigation strategy. These are programs that the FDA can require for certain medications with serious safety concerns to help ensure the benefits outweigh the risks, and they're really designed usually around reinforcing medication use behaviors and actions to support the safe use of that medicine. More rarely, we may withdraw a product from the market if the benefits are no longer outweighing the risks. And then safety communications are another tool that we use frequently to communicate risks. You saw in the regulatory history, we also had communicated about this previously through drug safety communications, and did so as well after making some decisions about what to do with montelukast after 2017. So for this particular case, we focused really on a couple activities. One is re-examining the risk-benefit for each indication. We looked at the different labeling and what our labeling options were, particularly had a lot of discussions about whether this warranted a box warning, discussed different communication avenues and used some of those. We also discussed REMS, additional studies, and decided that maximizing the labeling first before using some of these other tools is where we were headed, and then explored some other avenues for getting the word on the risk out there. So I'll focus on the first three here, because that's really where the actions ended up being in modifying labeling and re-examining the benefit risk assessment for each indication. So here's a decision framework for safety labeling changes. We started with a potential safety signal. Now we're in this assessment place where we're considering different questions to inform the regulatory action. And with the context of these questions, we're thinking about the risk-benefit framework and the uncertainty of both the data, but also on what impacts the actions will have on patients in the real-world setting. So we can ask, what did we understand about the risk after reviewing all of that data? We understood that patients taking montelukast have experienced a wide variety of neuropsychic events, although the available evidence is limited and primarily driven by spontaneous reports. There was significant uncertainty in both, mechanistically and timing. There was some question on whether it reverses after discontinuation. And we also understood that there was significant off-label use and a lack of awareness of the adverse events. We had two camps of goals. One are product use goals and the other agency goals to consider. The product use goals really revolved around patient and provider awareness and monitoring of any potential risks and making informed decisions about using the products while not inhibiting access. From an agency perspective, we wanted to make sure decision-making is based in science to increase the understanding of adverse events and appropriately respond to stakeholder concerns. So this was actually a pretty challenging thing to balance. There is actually a perspective piece that was published about this that describes some of the considerations we had. And in my opinion, one of the more challenging issues that I've been involved with, the perspective piece came out a few months ago in the Journal of Allergy and Clinical Immunology, and it describes the decision-making process in more detail. We had a decision science team come and walk us through different options and what the uncertainties were and how to weigh them. And, pretty interesting debates we had internally about what actions to take. But after careful consideration of the available data and the options, and particularly the feedback received during the advisory committee, the FDA required a box warning and revised the allergic rhinitis indication to reserve use of montelukast for patients who had an inadequate response or intolerance to alternative therapies. The FDA concluded, although uncertainty about the risks remain, a stronger warning was warranted given the potential seriousness of the safety concerns. Right, we had adverse events that included suicidal ideation and behaviors. The intent of the box warning was to raise awareness among healthcare providers. The advisory committee really provided evidence that healthcare professionals and patients and caregivers are not aware of the risk of mental and neuropsych side effects despite the existing warnings and the prescribing information. And the benefits of montelukast may not outweigh the risks in some patients, particularly if the symptoms are mild and adequately treated with other medications. And in the review of and the reevaluation of the drug's benefit and risk, the therapeutic context was a really important factor. The treatment landscape since 1998 has evolved a lot, particularly for allergy products. We have a wide variety of alternative, safe, and effective medicines, many of which are over the counter. So that landscape changed from what it was 20 years ago. So all of this is an example of the challenges in bringing the data together for decision making, as well as how the information continues to develop over time. So here's the updated timeline for montelukast after the 2017 feedback from stakeholders. A few other terms were added to the label. We had our pediatric advisory committee meeting, and then a box warning and other changes were taking place, as well as some other communication activities around neuropsych events. So to go all the way back to the start and to reemphasize the importance of pharmacovigilance, pharmacovigilance is important for continuing to learn about the safety of the product. At approval, we know about common adverse events, and we may have signals of other adverse events, and these inform the initial labeling, our pharmacovigilance plan, risk management activities, and any need for post-marketing studies. And in the post-marketing setting, we learn more about less common adverse events and refine our knowledge of specific adverse events, understand the condition of use. And this is a really iterative process. If montelukast demonstrates anything, that we continue to learn about the product over time. So I'll stop there, and we'll see if I have triggered any questions.
Hey everybody, this is the time for our general Q&A for everyone. So I'd invite everybody, if we give everybody power to unmute, if you'd like to ask a question out loud, that would be fine. If you'd like to type it in the chat, that would be fine too. But this is the time we've reserved for a general purpose discussion of all the materials today. Thank you to all our panelists. Was really an awesome presentation, and thanks so much, Monica, for walking us through the montelukast use case. I think it's really important just to show the fruits of all the labor that we put into all of this to have a resolution. So anybody can unmute themselves at this time and ask a question. We'd be happy to take them. And I'll reserve the right to ask a question in a few minutes if we don't see any questions.
I can't raise my hand. Do you hear me?
We do. Please go ahead.
This is Trinjung from Sweden, from UMC. It's very interesting to listen to montelukast and neuropsychiatric adverse drug reactions. I have a question on whether the timing of drug intake could have an impact on this adverse event. If you take it in the morning instead of in the evening, for example, on the sleeping problem.
Yeah, I mean, unfortunately, when we looked at the data, the quality, or the granularity of the time of the day, there wasn't a particular pattern that emerged to us. But yeah, I could see how it's possible that the timing of the product could affect events like insomnia or some of the other events that were noted, but it wasn't something that we observed in our data.
Thank you very much.
Sure.
So while we wait for other questions from the audience, maybe I can ask a question, and this is to all of the panelists, particularly relates to standardized MEDRA queries, which I understand are trying to make use of sort of different collections of terms that wouldn't be inherent to the tree. And that sort of thinking about, maybe we could do a compare and contrast of that with the cluster analysis that you talked about, Nicholas, which is also trying to learn more from the data about sort of the concept of disparate, sort of terms. I think that one of the things that is true about a lot of the trees that we used is they aren't great for syndromic things that end up where there's like a little bit of something here and there and there, and it's all over the map. But perhaps the standardized measure queries and the cluster analysis sort of try to take a bite out of that.
I mean, I think they do different things. I mean, I think that the SMQs to me are more similar to what we tried to do in a data-driven way with the distributional semantics. So basically, it's a way to bring together different adverse event terms that may relate to the same term, but where each individual term may be sufficient to say that you qualify to be relevant for that specific case series. The cluster analysis, in contrast, is really what you're saying now. It's more actually one of these signs and symptoms alone is not enough, perhaps. I mean, you can have terms, of course, as well, like neuroleptic malignant syndrome, where it sort of implies that you have a range of signs and symptoms. That does mean you meet some case criteria. But if you don't, you may need to have sort of a CPK increase, rhabdomyolysis, a few different things that would mean if you have enough of them, and then this is a probabilistic model. We didn't go into the details of it, but it's trying to get to that point where we can say which of these reports are similar enough in terms of the entirety of the reported adverse events. I will say in terms of using SMQs for signal detection, we didn't try that. So we looked at different levels of the hierarchy of matter, but also at the SMQs, and even the narrow SMQs oftentimes I think for signal detection are perhaps too broad. They may be very useful for building your cases and bringing potential cases into the mix. But in terms of the signal detection, in the most cases, it cast too wide a net, because it was then enough to have one of those adverse event terms to be included, and that just grew the case series too much to a point where we diluted it. That was our experience.
And to build on that, Nicholas, we had the same experience at the SNQ level. Whenever you have an SNQ that has an elevated reporting, when you dive into it, usually that's driven by just one or two MedRA PT terms rather than a general concept. So it's really important, like we did one with anaphylaxis at a signal, but when you dive into it, the bulk of the strength of the signal were from non-serious terms that were related to the broadness of the SMQ.
Yeah, I find that we often may start with an SMQ as a place to start, and then we start pulling out terms that, maybe have pulled in too much noise, but, it's nice conceptually to draw in the potential pool of cases. But yeah, I agree with what others have said about, the broadness of sometimes SMQs to bring in some noise.
Folks, it looks like we have a question from Tarek, old friend. Is there a quantitative tool to help in signal detection from clinical trials data during the actual development of the clinical trials, as opposed to just at the end when the package is received by a regulatory agency?
So this is not my area of expertise, but I think in terms of, clinical trials, there's the whole field of sort of adaptive clinical trials, right? Where this is the whole purpose is to set up a way that you can continually evaluate your data as you go along, and then possibly interrupt the clinical trial if it's enough. So I guess it would be sequential methods and so forth.
Yeah, Tarek, I know of one paper that that came out of, I believe it's France, but I have to look at it, where they used the tree-based scan statistics or the likelihood ratio methods during the clinical trial. It was a vaccine trial, so they were aided by the fact that they had a huge population to start with, and they were looking for the development of vaccine-related adverse events during the clinical trial. I'm happy to send out a link. But I think that was several years ago. I want to say that was like in the last it's, 4 or 5 years old at this point, but I know of at least that one paper.
That would be great. Actually, the reason I'm asking is because the new FDA guidance is focusing on the aggregate analysis as one of the sources of signal detection during development, basically during the process, not after the fact. So that's why I was asking whether there are any aids to actually do this while data are still blinded in many trials, actually. So because the unblinding will create an awkward situation, and many companies are feeling against doing this, at least not on frequent basis,
And I can just add a little bit to that, although not on the pharma side, but our vaccines group has published on a method they use for observed versus expected during clinical trials. Again, not my area of expertise, vaccines, but I know they are using some quantitative methods for in-stream review of clinical trials data.
So do you plan to put it in the chat, Judy?
Yeah, let me take a look. I'm going to mute myself.
But you are on mute. Yeah.
Oh, I'm sorry. I was going to say, give me a few minutes to just check it out in PubMed and see if I can find the link. I'll put it in the chat if I can find it in a few minutes. But if not, feel free to privately message me your email and I can send it to you. We have a few minutes left, but just in case.
Okay.
Folks, we'll give another couple minutes just to see if there are any lingering questions. Additionally, I want to invite everybody to be sure to fill out the skills course evaluation. So the skills course evaluation, I think, is right now for all of the skills courses together. This is just one of them. So I think you can fill it out for any of the courses that you have attended, but we would appreciate any feedback that you have for this particular course, both in the format, the material covered, if it was at the right level. And so I'm going to post right now in the chat the link that you all should have received in your email anyway as a skills course attendees. But just to remind everybody, if you have a chance to do that, it's really helpful to us. We would really appreciate the feedback. So thank you for anybody who's able to take the time to fill that out. Tarek, I received your email, so I'll go ahead and send that out to you a little bit later because I'm having trouble finding the exact reference right now. But I think just last call for questions, and then I think we can give everybody some time back. We're supposed to end by 11:45 anyway, so that people can get to the next skills course if they'd like to at noon. Okay. Thank you all for joining us today. It's been our pleasure, and thank you to the panelists for presenting today. I really appreciate it. And we really, really hope to see you next year live in person at the next ICPE meeting. Thank you.
Bye.
Thank you.